Okay, so welcome to the REU Virtual Symposium. This program was started by the Provost Office at UGA under Karen Holbrook and the Provost Office at Clark Atlanta University under Provost Harris. The program has been running since 2000, and it's been supported in that time by the National Science Foundation, by Sally O'Connor, who is joining us as well as part of the audience. And we've been in existence long enough that some of our older graduates are now in faculty positions around the country. So we've seen a complete generation of you go through the program. Almost 91% of the participants go on in science, and about 46% go on in higher degrees. And it's particularly appropriate that Dean Dorsey should introduce you all because he, for many years, ran his own REU site in Florida. And so he has also been a strong supporter of this program. And Kelly Happy, who has been doing the bioethics, that's courtesy of the Franklin College of Arts and Sciences. And it's my pleasure to introduce Dean Alan Dorsey. He is the Dean of Franklin College of Arts and Sciences here at UGA. And I guess the oldest state chartered university in the US. Thank you.
Very good. Well, good morning, everyone. Thank you for the opportunity to meet with you for a few minutes this morning. I want to start by, first of all, congratulating all of you for completing the REU program this summer. And also I wanted to thank Dr. Arnold for and his colleagues for the work that they have put in making sure that this program could continue under these highly unusual circumstances. As Dr. Arnold mentioned, I'm a big supporter of REU programs and in general undergraduate research experiences. And in part, that stems from my own personal experience going back many years. When I was a young person, actually in high school, I was given the opportunity to work in a laboratory, what was then called the National Bureau of Standards. It's now called the National Institutes of Standards and Technology. And I started doing that while I was in high school and continued through my university days, working during summers and so on at this national laboratory. But it really gave me a kind of great insight into what it was really like to be an actual scientist. You know, it's one thing to take classes and to go into a laboratory class where you go into a laboratory class, let's say it's organic chemistry, you're given instructions on how to carry out the experiments. And under most circumstances, things work out. You write up your results. Going into a real scientific laboratory, it's a little different because you have no idea whether the experiments are going to turn out the way you want. That's part of science, is conducting the experiments, looking at the results, troubleshooting problems, advancing new ideas, rejecting certain ideas. And that's why it's so important, I think, to have these authentic and genuine research experiences for you to get a real understanding of what it's like to be a scientist. And so I would just say that experience for me was very formative in my future career choices. And I then went on and went to graduate school, got a PhD, was a professor of physics at several universities, and now find myself as the Dean of Arts and Sciences here at the University of Georgia. But I want to make sure that I support programs like this one to offer opportunities to the next generation of our nation's scientists, and that would be you. So I hope you found your work this summer to be meaningful. I know it can be exhilarating sometimes when you make a new discovery or have a new insight. It can be very frustrating oftentimes because you, as I said, you embark on an experiment and it just doesn't work out the way you thought it would work out, or it takes longer than you thought, or there were parts of the experiment or the study that you were doing which you just had not anticipated. But that's all part of the process of discovery, and I think it's important to really understand that. Again, I wanted to congratulate you. I know that many of you will be considering your postgraduate career options, and when you do that, I certainly hope that you'll keep the University of Georgia in your plans. I think that's, we have a lot of great programs here at the University of Georgia in the sciences, both in Franklin College of Arts and Sciences, so in departments like the genetics department, but we have other allied life sciences departments as well. But there are other programs across the university in science and engineering that might offer great opportunities for you. So when it comes to that point, if you're considering postgraduate options, and if graduate school is one of the things that you're considering, I would like for you to consider the University of Georgia as one of your possible options. And I'm sure you can always get back in touch with Dr. Arnold or others to point you in the right direction if you're considering graduate school options. So again, thanks for your participation in the RU program this summer. I know you have a full calendar this morning of your presentation, so I hope this all go well. And I wish all of you a great future career. Thank you so much.
Thank you so much, Dean Dorsey. And as I said, it comes from the heart because he has run an RU program just like this one. So
Bye,
our
everyone.
first, bye. So our first speaker is Nick Putney. He is from SUNY Plattsburgh in Upper New York. He is a biochem major. He is worked this summer and will probably continue to work with Dr. Ying Zhu in the Institute of Bioinformatics and Biochemistry, and he is going to talk to us about cancer incidence rates and trying to explain them in terms of the expression of the human immune system. Nick, it's all yours.
Okay, let me just try to share my screen. Minute.
He's coming up. There you go.
And you can see it now? All good?
Yes, we're good. Good.
Okay, let's see if I can back the slide, put it full screen. Okay, so hi everyone, my name is Nick Putney. As you mentioned, I am a biochemistry major from SUNY Plattsburgh, and my project for the summer was understanding cancer occurrence rates across different organs. So cancers of some areas of the body occur much more frequently than others. And these differences even exist when looking at organs that are somewhat biologically similar. So if we look over here at the small intestine and the colon, cancer of the colon is about 8 times that of cancer of the small intestine. We don't really know why this is, although we have had a few groups who have tried to understand this. So one such group was Tomasetti and Vogelstein of Johns Hopkins University, who found correlations between stem cell divisions and cancer risk. So if you look at the plot here, the x-axis is the total stem cell divisions, and the y-axis is the lifetime risk. And as you can see, total stem cell divisions are a pretty good indicator of lifetime cancer risk, but it's not perfect, and there are certain organs that are significantly higher or lower than predicted. And just looking at the correlation coefficient, so for those unfamiliar, this R value, you can almost think of it as a measure of accuracy, where 1 is the highest. So we're trying to better explain why some are higher or lower. So our approach. So we looked at gene expression data from non-cancerous tissue that was found in the Genotype Tissue Expression Database, which is a project undertaken by the Broad Institute of MIT and Harvard to get gene expression data for tissue samples. And then hypothesis-driven data analysis. So essentially, we formulate a hypothesis, And then through analyzing data, we tried to test it. And our hypothesis. So we hypothesized that differences in the cholinergic anti-inflammatory pathway are the reason for differing cancer occurrence rates among tissues. And this is just kind of a brief overview of it, and a little bit more about why we think this is a reasonable hypothesis. So there was a paper published in 2007 in Immunobiology where the authors claim inflammation is associated with most of, if not all, tumors and supports the progression. This is becoming a more widely accepted view of inflammation, that it plays a critical role in the onset and progression of cancer. And just a few, briefly, kind of the mechanisms behind how inflammation leads to cancer. So with chronic inflammation comes increased angiogenesis, which is just the creation of new blood vessels, increased cell proliferation or increased cell division, and tissue remodeling. And all three of these things lend itself to creating a tissue microenvironment that leads to cancer progression, or at least makes it easier to lead to the onset of cancer. And then a little bit about the cholinergic anti-inflammatory pathway. Essentially, the cholinergic anti-inflammatory pathway just refers to a neural pathway where vagus nerves release acetylcholine, a neurotransmitter, which then stimulates receptors on certain cells. And although the exact mechanisms aren't understood, this effect has been shown to have widespread effects on suppressing inflammation. So the idea is if inflammation causes cancer and this pathway can suppress inflammation, it's a good avenue to look at to see if it potentially can explain cancer occurrence rates. So a kind of workflow for the analysis. First, literature review to determine genes to be used in analysis, and this was initially genes involved in the cholinergic system, some more downstream genes. Then we examine correlations between the genes and cancer occurrence rates, and this provides us more insight to which genes may be significant in determining cancer rates. And then we built a regression model using gene expression data from those genes. And this is just kind of formed the regression model type. And for those, again, unfamiliar with what a regression model is, you can almost think of it as a line that best fits the data. So kind of just looking at this just example chart here, this line here would be the regression model that's constructed from these points. And the closer these points are to the line, you'd say the better the model fits the data. And then lastly, we tested the predictability of the model on tissues. Okay, so here are the results using the genes from the cholinergic system. This plot to the left here is just showing how each gene is correlated with cancer rate. So down here are genes that are negatively correlated with cancer rate, and genes directly involved with the cholinergic system are found to be negatively correlated with the cancer occurrence rate, which kind of falls in line with our hypothesis. And then this plot right here is essentially the distribution of values predicted from the model. And then we took the peak of each of these distributions, where each distribution represents a different tissue. And then we plotted it on the x-axis over here against the cancer rate. So, yeah. So while you can see there is a general trend where organs that have higher cancer rates are being predicted higher than those with low cancer rates, the points are pretty far away from the line, which kind of indicates that it's maybe not the best. And again, looking at this R value here, the correlation coefficient, it's a little bit lower than the model shown in the beginning from the other group. So we thought we could do better than this, so we revised our hypothesis to include genes more generally involved in the immune response. And some of these new genes included genes that code for cell adhesion molecules, genes involved in different neural pathways, various transcription factors, different growth factors, and inflammatory cytokines, which are just signaling molecules, basically. And this picture here just kind of illustrates the complexity of the immune system and how the nervous system plays a central role in controlling it. Okay. Once again, very, very similar to the results, same like plots from before, except this time we used 79 genes that were generally involved in the immune system instead of just in the cholinergic system. And in addition to the cholinergic genes, it was found that adrenergic receptors, certain interleukins, certain lymphocyte transcription factors all negatively correlate with cancer occurrence rate, and this indicates that these may possibly play a role in suppressing the onset of cancer. And then various cell adhesion molecules, certain cholinergic receptors, certain transcription factors, certain growth factors, and interleukins are positively correlated with cancer occurrence rates. So this kind of indicates that these may play a role in promoting the onset of cancer. And again, similar to before, this shows the distribution of predictive values for each tissue, where each one of these kind of distribution shows a different tissue, and that the peak of each of these is plotted on the x-axis and the cancer rate on the y-axis. And as you can see visually, it's like the points are much closer to the line, and this correlation coefficient is pretty close to 1, which indicates that the model does a pretty good job in explaining
is he back
the
on?
different cancer rates.
Just please keep going.
Okay. So, yeah, so the model using a wider variety of genes involved in the immune system greatly improved the predictability of the model. It's kind of a little more about what this means. So the results suggest that much of the variation in cancer rates among tissues can be explained by differences in immune response, including control of leukocyte migration and lymphocyte differentiation. So this is kind of making sure you have the right immune cells and immune cells get to the right spot, and along with norepinephrine signaling and certain receptors involved in cholinergic signaling. And these help play a role in making sure these processes are turned on and off at the right times. And kind of these have been shown through the analysis to maybe play a role in determining cancer rates between organs. So as was kind of shown before, while the model was good, it wasn't perfect, and certain organs were definitely higher or lower than predicted.
Is he back on?
Indicates that other biological factors may also contribute to the variation in cancer occurrence rates among tissues. Two different avenues that we may want to look at are oxidative stress and energy metabolism, which have both been shown to play a role in the progression of cancer. And then why is it important that we understand the different cancer rates between organs? So understanding why certain tissues are more likely to get cancer than others can provide insights into methods for cancer prevention. So the idea is if we better understand what's happening at a more mechanistic level when we go from normal cells to cancer cells, we can then target those specific areas and then create treatments for that. So future work. So as kind of touched on before, I'd like to explore other processes that will improve the model. I'd like to account for differences in cancer rates between males and females. Account for changes in cancer rates as people age, because there are certain cancers that predominantly occur in children and are pretty rare in adults, kind of vice versa, where there are certain cancers that occur almost only in the elderly and you don't really see in pediatric cancer. And we don't really know why. And then, so the data for this analysis was only from the United States. So we'd have to kind of test our model, at least the gene set, on data from other countries to see how well the results generalize. So conclusion, while future work is needed, this analysis is a step forward in characterizing the underlying reasons for the variation in cancer rates among tissues, which is important for better understanding the origins of cancer, to lead to better cancer prevention techniques in the future. And then, so just a few people I'd like to thank. Dr. Arnold for putting this whole thing on, even with kind of everything that's going on, and definitely making the best of it. My mentor, Professor Zhu, who was, I learned so much from this summer and continue to work with, hopefully. The National Science Foundation, because they're the reason I'm able to be here. And then Dr. Tyson and Dr. Miller from my home institute for kind of guiding me through the process of applying to the REU. And then a few others who I emailed many questions. We're always happy to help. So thank you to them. And that's all I have. Thank you.
Thank you very much. So there's a little box, text box right below your video. You're welcome to send in questions. So and while you're doing that, I'll just ask a question of Nick right now. So how many genes did it actually take what percentage, were there a few genes that explain most of the variation in incidence rates?
So it's hard to give an exact answer to that, but So there's the final model used, we had 79 genes here.
Uh-huh.
So I mean, we could. I haven't explored it much, but you could explain most of it with significantly less genes. But the idea is, I thought that adding, for the most part, the actual biological scope of these genes isn't that large. So including more genes involved with the same biological processes, we thought better supported our kind of hypothesis.
Okay. Okay. So, and how much do you think if you look at the age of the people that will provide more explanatory power in your model?
So I don't know if they necessarily provide more, or I guess, so most likely I would say these kind of results may not expand across age groups, because, like, for example, in pediatric cancer, cancer that arises from the epithelial tissue is not very common. That's predominantly the type of cancer in the more elderly population. So I don't think this will kind of transcend age group. So I think we would have to actually look at different processes and different genes to explain that.
Okay. Yeah, so I think that's a good direction for you to go in, just because the immune system is age dependent. It doesn't work so well in me, but it works pretty well in you. So that's probably a good thing. Well, thank you so much. I will now move to the next speaker, which is Ashley Artist. She is from Norfolk State University. She's pursuing a biology degree there. She worked with Dr. Baptista on Cryptosporidium. That's a small eukaryotic pathogen that has a very small genome, and she's looking at causes of its virulence, how it gives us trouble. And it's my pleasure to introduce you, Ashley. Please go ahead.
Hello. Let me just share this.
We got it.
Everybody see me? Everybody's good?
Yes.
Okay.
Good.
Hello. My name is Ashley.
I don't think we're sharing our screen because I don't see us on the Monumental Media.
So, I see the screen. Keep going. Yeah, I think the screen's there.
Okay. Well, my name is Ashley Artist. I am a senior at Norfolk State University, and I will be presenting on discovering genomic variants and virulent strains of Cryptosporidium. So first, what is Cryptosporidium? It is a waterborne protozoan parasite that infects the intestinal and gastric epithelium of vertebrae that will then result in diarrhea and abdominal pains. So during its life cycle, it contains different life forms. For example, it starts off as a sporozoite. Then after it embeds in the epithelium, it turns into a merozoite. And then after that, it goes into a microgamete and a macrogamete, which are male and female, which means that they can mate. However, it's difficult to work with this parasite in vitro because it dies before it can actually reach a sexual stage. And there's only one FDA-approved drug against Cryptosporidium. So this is a disease that affects everybody. So cryptosporidiosis is the second leading cause of all waterborne diseases, primarily in immunocompromised hosts and infants. So here I have two different pictures. One is of a little girl. She's walking the streets of Bangladesh where they do have open sewers, which makes their people more susceptible to infection because of, you know, infected animals and their droppings. However, I do have also a picture of just children playing in public pools, and that's because chlorine is not effective against protecting people of the effects of Cryptosporidium due to their encasing that surrounds the oocyst. So, like I said before, Cryptosporidium has a broad spectrum of hosts. For example, the first known species associated with cryptosporidiosis were C. mirus and C. parvum. In the 50s, C. melagradis was associated with the morbidity and mortality of turkeys. In the 70s, C. parvum was associated with bovine diarrhea and neonatal diarrhea in young calves and lambs. And finally, C. bailey was associated with respiratory diseases in poultry, game, and birds. So why is it important to study this parasite? Well, Cryptosporidium easily infects animals that humans encounter, which has been concluded that public health and economic standing will be put at risk if this parasite is not properly investigated. And also cryptosporidiosis is considered an AIDS-defining illness, and the disease because it is a disease prevalent among the immunosuppressed. And also discovering and analyzing the genes of this parasite can aid in determining the severity of infection the strain is capable of. For example, like I said, it could the virulence the virulence, it can range from asymptomatic spread of oocysts, such as sneezing, coughing, or breathing, or it could be life-threatening disease. So what was my project for the summer? I compared 4 strains of C. parvum in order to characterize variants such as SNPs that will indicate which strains are more virulent than the others and determine variant gene candidates that can be tested for virulence factors. So what is a SNP? A SNP is just in different genomes of different people, a single nucleotide chains. As you can see, the purple person has an A, the blue person has a G, and the orange person has a T, but it's all in the same space in the genome. And like I said earlier, more virulent versus less virulent. Less virulent is just the asymptomatic spread of oocysts through sneezing, coughing, or just breathing, and it has less of a symptomatic effect on the body, whereas more virulent strains are more debilitating. So what did I do for the summer? All of these are my methods. It's a pipeline of my methods. So I was given 4 raw Illumina reads of the 4 strains of Cryptosporidium, which were bunchgrass, sterling, Washington, and INRA. These 4 strains were then compared to the reference genome of C. parvum, Iowa ATCC. And once those were compared, a GATK file was then prepared. And what that is, it just reveals all variant candidates that was analyzed. And after that, I had to filter these by mapping quality and depth. And then after that, I used SNPF to annotate the SNPs that were found. And all of these methods were used, like I had to learn how to access the cluster at UGA, and I also had to do coding. So in the end, I had over 4,000 genes, but only 540 had at least one variant called. And the comparison of all these genes collected showed that Washington and INRA were most divergent from these samples compared to the reference. And the 40 genomes that they share will be analyzed to look for other virulence factors. So these are a couple graphs. This graph right here are the 40 genes that were spread amongst the genome. And as you can see here, they're most prevalent on chromosome 5. And so I did this by submitting my candidates on CryptoDB to find their molecular function and metabolic pathway. Out of all 40 genes, 14 were uncharacterized and have no molecular function. And as I said before, the variant genes are mostly concentrated on chromosome 5, and some of these genes are responsible for protein synthesis. So in conclusion, since Washington and INRA are more virulent than bunchgrass and sterling, the 40 shared gene variants could be the starting point to check for virulence factors in Cryptosporidium. These genes were found to be mostly concentrated on chromosome 5. However, further characterization of these genes are needed. And there are 25 virulence factors that are previously identified in Cryptosporidium. These factors are related to host pathogen interactions such as adhesion, locomotion, invasion, and proliferation. And some host factors are associated with variations in both severity and risk of infection. So the 40 candidates that I found will then be compared to the 25 virulence factors to find any matches. And virulence markers can further be used for molecular diagnosis, and the unique genes, like in this slide, INRA had 176 unique genes, and Washington had 138, and these will be used to these will also be analyzed to find virulence factors. So here are my acknowledgements. I just want to thank Dr. Arnold for accepting me and allowing me to benefit from the RU program at UGA. Dr. Baptista, who was an amazing mentor. I feel like we worked day in and day out to get this right. And also Dr. Kissinger. She really encouraged me and really helped me throughout this process and kept it all together. So Thanks, guys.
Is the plan to, first of all, people in the audience, you can submit questions in the little box right below the video, and I'll present them for Ashley to answer, but I had a question of my own. And so what was the number that was in of virulence markers that were in common between all the 3 strains? And are those the ones you would go after if you were interested in, trying to stop the problem?
Well, first, we wouldn't necessarily go after, let me just go back to this. We wouldn't necessarily go after, because all of them in total have 21, and that was to be expected, 21 shared genes, and that's to be expected because they're all the same strain, the same type. However, INRA is from Europe, was a European sample, and Washington is from the US, and these are the most divergent samples from the original reference. So we would use the 40 that is shared to see a commonality amongst the two, because to also factor in geographical standpoints. So that's why we would start with these 40 to characterize and then spread out.
Okay. And so let me see if there's any more. Oh, I've got some, let's see if we have some questions here. So of the ones that you were thinking about, in fact, targeting that set that was in common, are any of those annotated already that are known virulence, associated with known virulence genes?
No, not yet. They have to be ran through. We know that 14 are uncharacterized and have no molecular function, but the rest of the 40, it's not done yet.
Okay. So I have a question here for you. I'm just going to highlight it and read it to you. And I'll put it, it should be on the screen. Does knowing virulence help with treatment of Cryptosporidium at all, or are they mainly useful for diagnosis?
They're actually really helpful for both. So I highlighted bunchgrass and sterling in green, Washington and INRA in red, because these 2 are more virulent and these 2 are less virulent. And knowing virulence can help in diagnosis because it helps with knowing how severe the infection can be, or how severe the diagnosis could be. But it also helps in drug discovery, because once we know the genome and we know how it attacks the host, we can counteract that through drug discovery. But it helps with both evenly. It's a 50-50.
Okay. Thank you very much. So I think what I will do is now, Ashley, move on to, and thank you so much. It was a wonderful presentation. Our next speaker is Olivia Asher. She's from SUNY Lehman in New York City. She's a biology major. She worked with me this summer, and she explored one of the oldest symbiotic relations on planet Earth between land plants and their root fungi. That's a symbiosis that's existed for at least 400 million years. And she's going to look at the population ecology of this interaction. It's my pleasure to introduce you, Olivia.
Thank you. Hello, everyone. Just going to share my screen. So thank you for the introduction, Dr. Arnold. My name is Olivia Asher, and today I'm going to be presenting Who Will Survive? Ecological Modeling of Nutrient Exchange Between Plants and Arbuscular Mycorrhizal Fungi. So before I get too deep into the project that I worked on this summer, I'm going to define for everyone what arbuscular mycorrhizal fungi are. So if you look at this picture here, you'll see a plant root. And inside of it, there's a bunch of small dots, one of which I've highlighted here in red. So these dots are arbuscules formed by the arbuscular mycorrhizal fungus. And these arbuscules are sites of nutrient exchange. So plants will provide mycorrhizal fungi with carbon, which the fungus needs to grow. And in return, the fungus will provide the plant with nitrogen and phosphorus. The plant is not as good at absorbing nitrogen and phosphorus from the soil as the fungus is. So this symbiosis is beneficial to both parties. So when you have a successful mycorrhizal collaboration with a plant, it can actually help plants grow both bigger and stronger. They will be more resistant to pathogens and their biomass will be increased. So it's important for researchers to understand this relationship between the fungus and the plant, both to help understand ecosystem health and to understand how to improve plant growth in an agricultural setting. So for our study, we want to create a model, a mathematical model that would describe the relationship between one plant and two different species of fungi. And we want to look at how these three organisms would exchange the nutrients, carbon, phosphorus, and nitrogen, as well as looking at the competition between the two species of fungi for resources. Oftentimes, one plant will collaborate with many, many different mycorrhizal symbionts. However, very little is known about how these symbionts interact with one another. So that's why we wanted to focus on looking at the competition between two different types of fungal symbionts in our model. So a model, just to be clear, is sort of a description of a system, but it also allows you to make predictions about how the system will work given different conditions, different soil conditions, and different types of fungi and plants. So to make a model, the first thing that you do is to learn about an interesting system. So in our case, we've already done that, even just in the course of this presentation. We learned about the two fungi, the plant, and the nutrients they exchange. The next step that I did in this internship was to develop equations to describe this system. So in this case, I developed ordinary differential equations based on the Lotka-Volterra model. So the Lotka-Volterra model is a model of an equilibrium model that describes how two different species will compete for resources. And based on this historic, well-established model, it shows that even if two species are competing for the same resources, they can still reach a stable equilibrium where both species survive. So we took that model and applied it to our two fungi, and then we added in new equations with different components. So different equations to describe nitrogen, phosphorus, the plant, rather than just the two fungi. So our next step was to see if the model was working. So we used MATLAB, which is a mathematical graphing program, to show what was happening to our species over time based on different initial conditions. And we wanted to see, was everyone surviving? Was the plant surviving? Were the two fungi surviving? And for most of our models, the plant died. And it did not survive. It was not a good model. So we made around like 12 or so models before we finally got one where it did indeed reach an equilibrium condition. And then our next step was to say, all right, so we see that the fungus is reaching equilibrium with the plant, the two fungi are surviving, the plant is surviving, it's all great. But is this equilibrium stable? Will it always try to go to equilibrium no matter what the starting conditions are? So then we use a stability analysis, which I'll get into later. We use an isocline analysis to show that our model was indeed stable. So the next step of our project, which will be done after the duration of this internship, would be to start using the model to fit experimental data, to make predictions, and to explain results. So the cool thing about systems of ordinary differential equations is that you can display them graphically. So I'm going to take you through a sort of diagram of the way that we model the system so you will understand what we did there. So if you'll remember, our system, what we wanted to do with our project was to describe the relationship between one plant and two different species of fungi, which were collaborating with the plant, but probably competing with each other. So the first thing that happens in the system is that the plant produces carbon using photosynthesis. So the red circle here represents the reaction, and the white square is what is produced by the reaction. So this is photosynthesis. Next, the fungi take carbon from the plant. So they're influencing the plant to give it carbon in RC1 and RC2. And then this carbon that they've taken from the plant, they then use to help with their own growth. So that's the GF reaction. Now, the two fungi are also competing with one another. So that is this term right here, A alpha beta, which is a competition coefficient, an idea taken from the Lotka-Volterra model that describes the effect these two species have on one another. And the two fungi can also die. So that's the death terms right here, D1 and D2. Now we're going to go to the soil. So the fungus is taking nitrogen from the soil in the reactions RN1 and RN2, and it's changing this nitrogen into a biologically available form right here, NM, which is then transferred to the plant in GP and used to grow the plant. And the same thing happens with phosphorus. So phosphorus is taken from the soil, turned into a form that the plant can use, and then used to help grow the plant. So now we've made this model. Our next step was to check that our plant and fungus survived, which I mentioned before. So we did that with MATLAB. So once we put in our equations into MATLAB and graphed them over time, so we have time, this is just arbitrary time, arbitrary concentrations. The one thing that we care about here is the shape of the graph. So you'll see right away that it looks like the plant and the fungi are surviving and they appear to be reaching a stable level. So that's great. It means our model is behaving how it wants. The next step that we did was to compare the predictions of our model to what had been previously observed in experimental studies. So one study by Hammer et al. 2010 predicted that when plants hoard carbons, that they do not share carbon with their fungal symbionts, the fungi in return will prevent the plant from getting nutrients like nitrogen and nitrogen and phosphorus. So you'll see here that in our last graph, the fungus was reaching about 10 arbitrary concentration of nitrogen, and now we're going up to almost 30. So when we made the plant hoard carbon, the fungi also hoarded nutrients, just like you would see in the real world. Another piece of evidence that our model is working. So the next thing that we did was examine what happens when a fungus is parasitic. So some fungi, instead of sharing resources with the plant, would just take carbon and return nothing to the plant at all. It's a one-sided relationship. So to test whether or not a mutualistic fungi would be superior to a parasitic one, we made fungal species 1 a parasite in our model, and we made fungal species 2 a mutualist. And you'll see right away that the mutualistic fungi is much more successful than the parasitic one. So our model predicts that mutualism is preferred over parasitism. Another thing you'll remember, our model is an equilibrium model. We see that the plant and the fungi are reaching equilibrium. So that's great, but now we want to check, is this equilibrium stable? So the way that we did that was to use an isocline analysis. So the orange line here is what happens to fungus number 2 when fungus number 1 is not changing. And the blue line is what happens the other way around. So what happens to fungus number 1 when fungus number 2 is not changing. And the point where these two lines cross is the equilibrium point. So now to tell if the equilibrium is stable, we pretend that we started with a concentration of fungi that's somewhere else on this graph. So let's say we started up here. Would the values of these two fungi return to equilibrium? So if we started with a very high concentration of fungus 1 and 2, they would decrease and go towards the equilibrium value. Where if we started with a very low value of fungus 1 and 2 below both lines, again, it would go towards the equilibrium value. Now, if we start over here where we have too much of fungus 2 but not enough of fungus 1, again, we're going towards equilibrium, and the same thing happens in the opposite way on the other side. So you'll see that based on the way that these two lines cross, it shows that our equilibrium is indeed a stable equilibrium. So our model has several benefits. It does reach a stable equilibrium, which we wanted, and it also favors a mutualistic relationship, which is hoped because we are looking at a mutualistic fungus. However, models are not real life, so they always have some drawbacks. So not all experimental results are portrayed by our model. For example, many studies find that if there's too much nutrients in the soil, too much nitrogen and phosphorus fertilizer is actually toxic to the fungus and plant. Our model doesn't have a mechanism to account for that, so we don't see that. Also, in the real world, nitrogen and phosphorus are very different elements, and they're absorbed differently by the fungus and related to the plant differently, but our model doesn't account for that difference. So in the future, we would like to continue improving our model by possibly adding in, the differences between nitrogen and phosphorus or mechanisms for toxicity. And we also want to examine the equilibrium better and just learn more about what our model can portray. And in addition to building the model more, we also have an experimental side to the study. So one of our collaborators, Gia, will be doing quantum dot tracking to trace how these resources move in a real life system of two fungi and a plant. And then eventually we'll be able to combine these two sides of the study to have a very powerful mathematical explanation for our experimental results. Thank you so much for listening to my talk and to all the people who helped make this research possible. And let me know if you have any questions.
Okay, great. Thank you so much. So I'm going to wait here for a second for questions to come in. And can you talk about, are there both experimental challenges to tracking the carbon, nitrogen, and phosphorus in the plant and the fungi, and where can it be done now, and where do we need new developments? And that's one question I have.
Okay.
Yeah.
So again, Dr. Arnold is my mentor in the project, so I feel like he just wants me to share some more fun information with everyone. So when I spoke to Gia about what she was going to do with the quantum dot tracking, she said that right now she's working on like getting the imaging kind of stuff ready. And I believe that quantum dot tracking has not really been done for carbon before, and it's not been done when there are multiple species of fungi. So there is quite a bit known about how a plant and a fungus exchange resources when it's just one fungus and one plant. But when it's multiple fungi, that's where we don't know. And in the real world, there are many, many different fungi collaborating with one plant. So that's why this research is very valuable, because it's going to help us learn more about what's actually happening in the real life system, as opposed to just what we think is happening because of one plant, one fungus.
I have a question from the audience.
Okay.
And does let's see, where did that go? Oh, heck, I lost my question. Oh my, I did something, it's something, archive. I don't know, oh there, archive. Will you now be focused more on the model building or experimenting? Which are you going to be doing in the future?
Well, I am not at the University of Georgia, so I will definitely be working on the theoreticals of the model side. And there are collaborators, so I mentioned Gia is one of our collaborators at UGA who will be doing experimental. She's a chemist. She understands the experimental side. So I will definitely be more of the theoretical person here.
Okay. Let me see if I have anyone else. I think that is it. So what I will do now is we're almost on schedule. And thank you so much, Olivia. And it's now my pleasure to introduce Vaibhav Shah. He is a major in computer science. He's from Amherst College. He is looking at ORF1A and SARS-CoV-2. That's the target of remdesivir, the one known treatment for COVID-19, and it comprises about 2/3 of the genome. And he is going to be looking at understanding the variation within SARS-CoV-2. And it's my pleasure to turn it over to Vaibhav. You're muted, Vaibhav. Okay. And you turned it on.
Am I no?
Good.
All right.
Good.
So you guys see the screen?
Yes. Yes.
So my presentation will be on my project on the origin of COVID-19. And I was trying to study it using phylogenetic tools and using those tools to understand how it attained its human virulence. And my mentors were Dr. Arnold, but more directly, Michael Scarrow, a PhD student under him. And so what is phylogeny? Well, it's the study of an organism's evolutionary lineage and its history of change over time. And so it relies on evolution, obviously, because you're assuming that all species arise from another species. And phylogeny is usually documented by evolutionary trees, and usually there's species trees and gene trees. And species trees tend to be broader and can be built from gene trees, and that's what this project was trying to do, is make a species tree in the end. And so that was what the end product was supposed to be. It's actually still being made, because the gene trees, like Dr. Arnold said, there was one gene, ORF1A, that was huge, and it took forever to run because of that. And I had to request a lot of resources on Sapelo to run it. But once we have the species tree, we're going to use it to look for patterns and also extrapolate from it to predict possible routes of expansion and how the virus is going to evolve over time. And so the methods used to get where we are, well, first, before I even joined this project, there were samples of the virus from all over the world and from many different organisms too. Like, for example, bats and pangolins were included too, on top of the human samples. And all these were sequenced. There were over 7,000 samples, I think. And so what the output of that was the entire genome of SARS-CoV-2. And my mentor, Michael, actually took that and turned it into a sequence alignment by basically taking out the genes of interest from that genome and aligning them across all the different subjects that we collected data from. And so this is where I come in. I was given the multiple sequence alignments and told to make a species tree. So as you can see in the middle, I had to make a gene tree to be able to make a species tree. And so the input format of the multiple sequence alignment was actually FASTA files. And so there was a bit of converting that needed to be done. And then I used mainly RAxML to transform the multiple sequence alignment into a gene tree. And the species tree is going to be made using MPEST, which I'll talk about later. So what is a phylogenetic tree even? Well, the basic structure is that there's all these nodes where lines meet up and they represent ancestral species. And as they branch off, that represents divergence. And so the ancestral species had maybe some of them were mutated or whatever, and they diverged into new species that were distinct. And it doesn't actually necessarily have to be species. The taxa themselves can be any variant. Like, for example, a gene can just diverge over time. And so the length of the branch does usually represent time, especially as you go from left to right. And all the leaves are usually at the end because those are the species that are currently existing, unless if they're not at the end, then they died out. But for most of the data we dealt with, that was not the case, or at least hasn't been yet. And then polytomy is just where there's more than two divergence events occurring at the same time. And usually it's thought of as not actually being accurate because there's usually a divergence event after the first divergence event that led to the two taxa, the polytomy. But we didn't have to deal with that. So how are the gene trees made? The multiple sequence alignment was taken in using RAxML. And so RAxML basically looks through the multiple sequence alignments for differences. And so every mutation is taken in and it's thought of as an evolutionary event where this divergence might have occurred. And so RAxML basically makes a huge matrix. And they used a substitution model to create the gene tree called GTR gamma. And if you think about it, there's a lot of ways to arrange the tree just based on the differences you see. And some might be more likely than others. And so where the ones that are more likely are going to be the ones that have fewer, where you have to traverse fewer common ancestors to get from one taxa to another. So that means more divergence events, which means there's also going to be more mutations over time. And so that's why bootstrapping was used to make sure we got the best tree. And so we actually made 100 gene trees every single time, and the gene tree that occurred the most was referred to as the most probable, and that's the gene tree that was taken in the end. And so the species tree is going to be made from the gene trees using MPEST, made by Dr. Liu from UGA. And this program is going to take in all the gene trees and basically take an average of them, because you're no longer just constructing from a multiple sequence alignment. You're looking at the gene trees and trying to find how the individual taxa arise from ancestral genes instead of using the other things. So multiple runs are going to be conducted again, because there's also multiple ways to make species trees from gene trees. And we had 10 gene trees. And so there's many different ways to make a species tree. And this program made by Dr. Liu is going to take the most probable again. And so how is this all going to be used in the end? Well, the species tree is going to be analyzed to show the evolutionary track of SARS-CoV-2. And hopefully that can explain how the virus has been able to cross species boundaries. For example, it's been able to get from bats to humans, and it keeps evolving. So maybe we can even extrapolate from that and see what it's going to do in the future. And this whole, like, looking at specifically what things might have changed, especially in ORF1A and the genes associated with its reception, could tell us how to treat COVID or even prevent it. And once we look at its evolutionary track, we can also anticipate if other coronaviruses are going to follow a similar path. So thanks for your time, and thanks for my mentor. I don't know if he's here, but thanks to Dr. Arnold too for organizing all this.
So thank you very much, Vaibhav. So we're with the different gene trees that you generated. Were they very concordant across the different gene trees? Were the trees look different?
They looked similar, but not identical. I think for the most part, the same, like, by location things were grouped together, which makes sense, but there might have been minor differences.
Okay.
I don't think they were exactly the same.
Okay.
And we're still waiting on gene S, so
right.
That's different.
And so how many differences were there in between the like 4,000 sequences that or 4,000 samples that you looked at in ORF1A? Were there how many different mutations would you see in the human lineages? Did you get some? Yeah.
Not exactly sure, but okay. Proximel outputted at least 20.
At least 20. Okay. Yeah. So it's a fairly early in its evolution then. In us, and I gather that the changes relative to pangolin and bat were more substantial. So they ended up at the root of your trees, I gather.
I know bat was I believe so. Yeah, bat was probably the root.
Definitely right. Okay, great. Well, thank you very much. And this we'll have more to the story in some more talks. There were several students that worked with VIBOF on this project, so with different focus. Thank you so much. And so now it's my pleasure to introduce Emily Moore. She's from Wofford College in South Carolina. She's a dual major in biology and computer science, and her mentor was Jan Mrazek, and she is examining simple sequence repeats in bacterial pathogens and what role they play in phenotypic switching. My pleasure to introduce you. You may need to unmute yourself. Yes. Okay. Good.
Okay, so like Dr. Arnold said, my name is Emily Moore. I am a biology computer science double major at Wofford College in Spartanburg, South Carolina. And this summer I had the opportunity to work and research looking at long simple sequence repeats in Helicobacter pylori. So a simple sequence repeat is a tandem iteration of a single nucleotide or short oligonucleotide. So that would be something like AAAA, TG, 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 and so on. And several characteristics of simple sequence repeats are that they are polymorphic in length, which means they are constantly changing their length. They can get shorter, they can get longer. So like in this example here, this repeat gets longer by 2 nucleotides, but it can get shorter. And changes like this can promote phase variation, which is the reversible inheritable switching of phenotype. So kind of like an on-off switch for a gene. So for example, if there was a simple sequence repeat in the promoter region of a gene, it could mutate and it could get longer, it could get shorter, and that can cause that gene to be turned on, turned off, non-functional, and so on. And so simple sequence repeats are usually found in eukaryotes and are generally rare in other bacteria genomes, but they are, however, seen in the genomes of host-dependent pathogens like Helicobacter pylori, which is what we were looking at. So Helicobacter pylori is the pathogen that is responsible for things like peptic ulcers, gastritis, gastric cancer, and it is really important for H. pylori to enter its host immune system and start adapting to that environment very quickly. That leads us to our question of how do these simple sequence repeats, or SSRs, contribute to the plasticity of H. pylori's gene regulatory networks and its adaptations to host environments. And from this, we hypothesize that this SSR-facilitated phase variation plays a very important role in H. pylori's ability to infect its host and quickly adapt to that environment. So to start by doing this, we started by downloading 180 H. pylori genomes, completely sequenced H. pylori genomes, and started looking at the long SSRs that were present in those genomes using different programs and online softwares. From this data, we were able to pick 8 genomes we believed were representative of all 180 H. pylori genomes, while also having a decent amount of long SSRs. And so what we mean by long SSRs can be determined from these graphs. So earlier I said that SSRs were generally rare in other bacteria genomes like E. coli, which we can see here. So this x-axis is the length of a repeat by nucleotide, and the y-axis is how many, like the number of those repeats at that specific length. So here, this black dotted line is the actual genome that we're looking at, and then these other lines represent random simulations using stochastic models and what would be expected. So as you can see, the black line really follows these random models, which means that most likely those simple sequence repeats just occur by chance. They're not really significant. And then you can see that once the repeat gets to about length 10, you don't really see any more repeats, which shows that the repeats aren't really used in these E. coli genomes. Now, when you look at that compared to Helicobacter pylori, you see that below 10, it follows those random model lines really closely, which is expected. Most likely those SSRs do not occur by chance, or they do occur by chance. And then once you get greater than 10, you start seeing some more repeats that are greater than length 10, so getting around length like 16, 17, 18, so they're really long. And it has been supposed that these repeats occur by, and they're maintained by selection. And these are what we're referring to when we talk about those long SSRs, which are anything that deviates from those random model lines. They would be unexpected. And then this chart shows the count of long SSRs we have in our selected H. pylori genomes. So these first 8 are our representative genomes that we picked, and then these these other 2 down here are different genomes that we found, and there weren't as many long SSRs present. And so this is just to show that there are some H. pylori genomes that do not have a lot of long SSRs in their genomes. And then our 8 that we selected do have a good amount. So after we determined which 8 genomes we wanted to use to represent all of our other genomes, we started doing a comparative analysis of the SSRs that we found, and started looking at which genes are they associated with and their importance. And so here's some examples of that. So in this first example, we have a repeat that is associated with 2 genes that code for outer membrane proteins. So these colorful rows up here are the actual DNA sequences for our 8 Helicobacter pylori genomes. And then these rows down here are the genes that are associated with them. So this yellow row here is the DNA sequence that is associated with the Helicobacter pylori J99 genome. And then that corresponds with this row right here. So this means in the DNA sequence, there would be a gene for an outer membrane beta barrel protein, and then 106 nucleotides downstream of that would be the repeat. And then 174 more nucleotides downstream of that, there would be another outer membrane beta barrel protein gene. And so the first thing to notice is that you do have some repeats that are the same length, like these that are length 14, but you also have some that are longer than that. And this kind of shows the variability of the length of these simple sequence repeats. They can get longer, they can get shorter, they're always changing because they're hypermutable. The second thing to notice is that this specific repeat is found in between 2 genes. So that means that they could possibly be in the promoter region of the second outer membrane beta barrel protein gene, or in or near that promoter, which it could alter that promoter if it was to mutate, which we can see that it probably does, and alter that promoter that could then alter that gene. And so we found that 18 out of 88 of our simple sequence repeats that we found were associated with outer membrane proteins, and outer membrane proteins are used to help facilitate the transfer of materials inside and outside the cell. So it interacts with that outside environment a lot. The second example is a dimer repeat. So you have the GA, so it's 2 nucleotides. And then again, you can see that variability in the length, meaning it's changing a lot. And so this specific repeat is associated with a glycotransferase protein, which helps with the transfer of sugars, and it even helps with the synthesis of lipopolysaccharides, which are found in the cell membrane of H. pylori. It looks like this repeat is in the gene of glycotransferase protein. So that means, again, if it were to mutate, it could alter that gene and put a stop codon, change the amino acid sequence, and so on. So 7 out of 88 of our SSRs that we found were associated with glycotransferase proteins. And it's very interesting, and we expected to see stuff like this because outer membrane proteins and glycotransferases and even genes that code for adhesions, which also work with the outside of the cell and interacting with that, are very important to H. pylori because it does need to interact and fight off that host immune response. So we expected to see SSRs associated with this because it will promote that antigenic variation, which is used to help combat that host immune response. One thing we did not expect, however, is to find SSRs associated with things like DNA methyltransferases. So it has been proposed that DNA methyltransferases actually have regulatory roles in Helicobacter pylori, which is atypical. Basically, the DNA methyltransferase would modify the DNA by adding a methyl group, which could then impact the gene expression, impact how that DNA sequence will interact with RNA polymerase and transcription factors, and so on. So if this DNA methyltransferase already has some sort of regulatory role where it impacts gene expression, adding an SSR to that could influence a lot more genes than just this one. So this SSR is specifically found in this DNA methyltransferase gene. So for example, if this SSR were to mutate and somehow make this DNA methyltransferase non-functional, it not only would impact this DNA methyltransferase gene, but it would also go on to impact all the other genes that this DNA methyltransferase would regulate. So going back to our question is, how do these SSRs impact these Helicobacter pylori gene regulatory networks and so on? And so one thing we expected to find, because it is seen in other host-dependent pathogens, is genes associated with SSRs that are related to structures present on the surface of the cell. So your outer membrane proteins, your adhesions, your glycotransferases. This is important to Helicobacter pylori because it does promote that antigenic variation, which can help fight that host immune response. One thing we did not expect to find was SSRs associated with DNA methyltransferases, which had been seen, proposed to have regulatory roles. And so adding that phase variation to this regulatory gene could influence a number of other genes, not just the gene that is directly involved with the phase variation. And then our last important find that we noticed was that SSRs vary in length, which proves they're hypermutable and they're very variable. And so from here, we plan on looking at the transcriptional start sites of these simple sequence repeats to look at, are they in the promoter region of these genes? Are they in the protein coding region of these genes? Are they at the start of the gene if they are in the protein coding region? And look at how, if SSR facilitated phase variation does occur, how does it impact the gene? And that's it. Thank you.
Thank you. Thank you very much. While I wait for questions to come in from our audience, which those questions can be put in the little box below the images, I'll just take my shot at a question. Where do the simple sequence repeats come from? How do they get generated, since they're so unusual in non-pathogenic bacterial genomes?
So they just occur. So like most of the time, like if you look at the human genome, there's a chart, I don't have it, but that graph that I showed you with the long simple sequence repeats,
right?
Genome, it goes all the way across. So it has a long simple sequence repeats. Okay. It is supposed that they're like maintained by selection because they're important for like regulatory and gene expression. And so they just occur, but they're maintained, or it's been proposed that they're maintained by like the natural selection, and they're beneficial. Okay.
So I was sort of curious that, it could be the repeats could get generated by sort of errors in replication, or maybe some form of recombination or something. That's what I was wondering about. And then, like you say, once they occur, then natural selection can take over. Do you have any plans for doing experimental work on the DNA methyltransferases that have these simple sequence repeats?
Not that I know of right now. I know the only plans that we have right now going forward is starting to look at those transcriptional start sites to see where they take place.
Okay. And also, where did the SSRs occur, at the end or the beginning of the gene they were of the methyltransferase?
So they all kind of vary. So it looks like these numbers, so basically in the gene, it's about 1,300 nucleotides in, there's the repeat. So it looks like in this specific DNA methyltransferase, it's near the end of the gene. But we're particularly interested in SSRs that occur like near the beginning of the gene, because if there was some phase variation that somehow caused a stop codon, then that would be kind of important.
Right.
So, but they all kind of vary.
Okay. I'm trying to, you just got a question from the audience, and I lost it when I moved it to the screen. Let me just see if I can find it again here. Let's see here. I almost, okay. I lost the question when I moved it.
I see it. It's why did I pick these strains out of the 180?
Yes.
So these were just the strains that we thought were the most representative of all the other 180 genomes.
Okay.
They also had a decent amount of long SSRs.
Okay.
I know a few of them were associated. It went away.
That's okay. But so you were going to say something about where these came from.
Four of them were actually like in the phylogenetic trees, like some of the beginning ones too.
Okay.
The most representative of all the other 180 genomes.
Okay. Well, thank you very much. We're going to take a brief break right now, a bio break, and we'll start again with Frederick Little at 10:30. I sort of jumped the gun with Emily Moore and started her at 10, and I apologize to the audience if you're just coming in. We will be putting putting these talks out on YouTube. So yeah, and also the same link immediately following the virtual symposium if you want to pick up again a talk that you missed or we had some technical difficulties with. You can revisit it, and that's good. So I'm just going to be here. If you got questions, I'm not going anywhere, and I'll try to pull in some of the our speakers so far. If you have questions that percolated up, I'll be glad to forward them to them to address them. Welcome back to the virtual REU Symposium. It's my pleasure to introduce Frederick Little. He is a major in bioengineering at Clark Atlanta University, and he worked with Dr. Casey Bergman at the University of Georgia on one of the most fundamental problems in biology, namely the transposons, mobile elements in the genome. And he's going to talk about his work on bioinformatics tools to characterize those transposable elements in genomes. Thank you, Frederick. All yours.
Good morning.
Morning. And
All right, good morning.
good.
I
It's full
am,
screen.
all right, perfect.
Good.
All right, good morning. I am Frederick Little, a senior dual degree engineering major from Riverdale, Georgia, currently attending Clark Atlanta University. And this summer I worked under Dr. Casey Berkman as well as Preston Basson on my project visualizing data from transposable element insertions in genome sequences. So what are transposons? Discovered in the 1940s by Barbara McClintock, they are DNA sequences that are capable of moving from one location in genome to another. They're highly repetitive, constituting more than 50% of the human genome, and are responsible for some human diseases. It is imperative that we want to better understand genetics as a whole, that we understand what transposons are and what they do in the genome. Here is an example of one way that transposons replicate. There are multiple. This is the cut and place method, where you see there's a donor strand. It's actually cleaved from it and then put into the target DNA strand, giving you the transfer strand. Genome bioinformatics. Computational biology is a very, very broad subject. Genome bioinformatics is the output of it. It is the study of whole genomes of organisms through the application of computational tools. The goal is to study large genomic data sets generated by high throughput DNA sequencers. And the availability of large data sets has stimulated the development of many computational tools for studying transposons. Among these are the McClintock pipeline. This is what we've been working with in the Bergman lab. So the McClintock pipeline is a computational tool used for the detection of TE insertions in whole genome shotgun sequences. Integrates data from multiple other bioinformatic tools. As you can see, there is a lot that goes on with the flowchart overall, but what's important to highlight is the amount of files that it actually produces. In addition to that, the types of files that it produces. So you're gonna have PET files, CSV files, PNG files, TXT files. To my right over here, this example is actually one of the CSV files that you may actually see from the data that's produced. But that brings about a question: how do we thoroughly visualize all this data, and how do we organize it? So our end goal: by building a template script in Python, we should be able to establish an efficient system to visually represent multiple data files from the McClintock pipeline. Materials online now. So GitHub is actually our notebook. Online kept everything together so that everybody communicated efficiently. Visual Code Studios actually put all of the different forms of Python languages that we were using for programming: Python, HTML, CSS, and Ubuntu was my operating system to run everything. HTML and CSS. As you may know, HTML is the standardized programming language for the World Wide Web. CSS, Cascading Style Sheet, is actually how you added the styles to everything, the visualization. That means colors, font, text alignment, size, border spacing, and layout in itself. Python is the two. Python is one of the main languages that is used now in coding everywhere. J2 is a template engine that's actually built into Python. What it does is it passes data from HTML templates, which in cooperation with CSS generates web pages. Over here, we actually see that from this McClintock run, we actually produce these files, different types, but we use this Python script to read these different files, which then fills in a generic Ginger template, and in cooperation with CSS, generates HTML files. Here is a very, very basic example of one of the earlier Ginger CSV templates that we worked with. So you probably know what an HTML page looks like on the web. Well, this is what does the heavy lifting, the code behind it, the bare bones and skeleton. You see these different values. These are the variables that get read into, and they then produce. So this is the file that actually does the reading itself. This is the Python Ginger template script that's charged with that. And after it reads those different variables, it passes it to the file. This is before and after on the left side. you actually have the JINSA template where you have the different variables. And on the right, you actually have actual file where everything's been produced into. As you can see, you have different variables. Some of the data has been read from the file itself, you can see. And the end result is going to be something like this, a web page that actually produces it in itself. Now, this is a very simple script used for the example itself, but our end goal is to have a script that actually visualizes all types of different files and is then very interactive to the point to where you can pretty much go to different pages, have navigational bars, and that's all accomplished with CSS, and a more complex script which is capable of reading all different types of files all at once. So there's no going back and forth. You can just run the script and it would read the entire batch that's actually produced from a McClintock run. Discussion. So we were able to establish an interactive visual representation of data as well as a structured way to navigate files from the McClintock run. We hope that in the future, we'll be able to make this system more aesthetically pleasing and simplistic. It's important that anybody can pick it up at any time period, and we want to make this something that is a mainstay for the entire pipeline. Acknowledgements. I'd like to acknowledge Preston Basson, the graduate student who worked with me day to day. He really taught me a lot about the coding systems and everything that we used. Dr. Casey Bergman, who actually set me into the lab, gave me those multiple goals to accomplish every week as I kept moving forward. And Dr. Jonathan Arnold, who really accepted me into this program and did all the work to put everything together for the program as a whole. So I really do appreciate everybody for that. Thank you.
Thank you very much, Frederick. It's a wonderful project. And okay, so I have the first question in. Are you manually adding in those data values, or that's their question. And I have to find where the question went. It seems I have, you think it would go near the top. Oh, yes. Yeah, so there's the...
So no, the McClintock pipeline actually is creating those data values. It's actually producing themselves. And that's all through the run, right?
Right.
What we're doing is we're creating a system that reads the files that have been produced from the McClintock pipeline and then visualizes it.
Right. Do you think it's very hard to go the other direction? So if somebody suddenly gets a graph of, say, where the transposable elements are in the genome, can they like zoom in or zoom out, do something interactive with the graphic? Is that capability built into the Ninja system?
It's possible. It's definitely possible. And that's something that would probably be built in later on down the line. Okay. It is a goal to make it as aesthetically pleasing as possible. But the biggest issue is, I would say, battling with making it also easy to pick up, because you want everybody to be able to just, jump into it.
Right.
But yeah, it's definitely something that we're looking to in the future.
So your goal is then to have a web resource so that if somebody wanted to look at transposable elements in Neurospora, or, some other organism, they could use this web tool to hopefully identify them. Is that sort of the make the McClintock pipeline widely available? Yes.
So, anybody could really understand the data that we have presented and make the data widely available. So.
Okay. Were you mostly focused on transposable the examples you worked with? Were they mostly in yeast, or were there? Did you?
Yes. Yeah, mostly yeast. Mostly yeast bioinformatics, and those transposable elements came from yeast genomes themselves. But yeah, the goal is after we understand that, hopefully after we understand transposable elements in yeast, we can then later on get to the human genome. But it's just so much right now that we don't understand that.
Right.
Using this is probably the best method.
Right. Okay. And do you this is going to? Do you feel this will help you with sort of your engineering aspirations so that projects you get involved in down the line, you'll be able to use these kinds of approaches in your future work?
Definitely. And that's something I definitely have to thank Dr. Bergman with, is tasked me with a project that actually appealed to the engineering aspect of my biology field.
Right.
And that's because, the whole point of engineering is efficiency, is to make something better. And yeah, it's important to study different things, but you always want to make sure you're doing it in the most efficient manner. And this really challenged me in my problem solving skills to just that, especially challenging my coding skills because they need a little brush up. So I'm glad.
Okay, great. And let's see if I have any more questions here. I think we're good. And so what we'll do, we just have a few minutes left, and just so that people coming in can will be on schedule. And but if we'll just give you a few more minutes in case someone would like to ask some additional questions. I was particularly struck that there have been several people from the two REUs that you've worked with this year that have focused on visualization. So Isaac focused on visualization of viral genomes. You're focusing on genomes with transposable elements, and then there was the visualization tools that Lily worked on for plant roots. So I'm sort of curious if there was any crosstalk between you guys on those different related projects this summer. And yeah.
Not particular. We didn't talk. It was more of us sharing our hardships. Like I said, you come into a problem or something, and encouraging each other. But we didn't really get too deep into it. I'm kind of interested to see what everything is up to them as well.
Right. So what do you think the most challenging part of the summer was for you?
So coding has always been probably the hardest aspect of everything when it came to engineering for me. So I'm glad that I actually got the opportunity to focus on it specifically and take my time to learn from it, and then also apply it to to biology, because something that's always happened is when I do take languages or when I do take whatever in school, I don't get the opportunity to actually apply it to what I want to apply it to. And I think that is partially the reason why it's been so hard. It's been such a struggle.
Right.
But yeah, it was a struggle at first to just understand and take in everything, learn to actually accomplish the goal. But it was rewarding. That we can say it was rewarding.
Well, thank you very much. And what we'll do now is get our next speaker ready. Our next speaker should be your colleague, Samari Massey, who is also will be coming on, I think, in a second. And hopefully he's unmuting himself, and also, yes, you're all there. Good. And again, whenever you have questions, so turned off. Okay. Yeah. So yeah, I'll keep going. Samari Massey is also in bioengineering and biology at Clark Atlanta University. He is working on molecular modeling of inhibitors of genes in Fusarium that affect denitrification, that is, production of nitrogen-related gases, and that's hopefully something that we can control and prevent greenhouse gases. And so it's my pleasure to introduce him, and this is what he will talk about. Thank you, Samari.
Thank you, Dr. Arnold. Can you see the presentation? All right, thank you. So, again, my name is Samari Massey. I'm a participant of the University of Georgia's RAU for Fungal Genomics and Computational Biology, and today I'll be giving a presentation on my process and results for molecular docking of candidate inhibitors to bacterial nitrite and nitric oxide reductases. So the aim of the research that I've been doing with Dr. Penniman is trying to slow fungal denitrification in the Fusarium verticillioides species of fungus. Denitrification is the reduction of nitrites or nitrates to produce nitrogenous gases, and we are trying to slow and stop this process for two reasons. First, fungal denitrification causes a loss in soil nitrogen, which ultimately reduces crop yields for farmers. And the second reason is because fungal denitrification releases nitrous oxide into the atmosphere, which is a potent greenhouse gas. In order to carry out this research, a computational screen of thousands of inhibitors was performed in order to gather a list of possible candidates that could be tested to bind to enzymes in the Fusarium verticillioides. However, the effectiveness of these inhibitors prior to the research was unknown. The enzymes they would be tested on are nitrite reductase, abbreviated to NIR, and nitric oxide reductase, which is abbreviated to NOR. Eventually, 25 candidate inhibitors were selected and were used in this research. Prior to beginning, we also hypothesized that these inhibitors may also bind to bacterial enzymes with the same function. In order to carry out this research, there were three main methods that were used. The first being functional annotation, in which we use peptide sequences of these proteins in order to not only predict what they do, but also made sure that we had selected the correct proteins. The second method was homology modeling, in which we used the same peptide sequences in order to predict the three-dimensional structures of these proteins. And the third method was molecular docking, in which we used 3D models of proteins and candidate inhibitors to predict where the inhibitors would bind to the proteins. In order to carry out these methods, there are also many materials or tools that had to be utilized. They are listed on this slide, and I will go over them briefly. Swiss Model is a 3D modeling software used to create possible representations of unknown structures. Pymol is a 3D model viewing and editing software. OpenBabel is a program and a library designed to convert between file formats that are commonly used in molecular docking. AutoDocVina is a molecular modeling simulation software that is very effective in protein ligand docking. Python and R are both coding languages. PDB stands for the Protein Data Bank and houses lots of information for 3D structures of proteins and other molecules. And NCBI stands for the National Center of Biotechnology Information, and it's a database that houses lots of information for other biology-related fields. Now I'd like to discuss the results that were produced from this research. First, I'd like to talk about my molecular docking results for fungal, NIR, and NOR proteins and compare them to previous molecular docking results that were produced by Dr. Penniman. On this slide, the black images represent previous molecular docking results, whereas the red images represent my own docking results. On the left side of the slide, you can see the previous and new molecular docking results of NIR homology models. In these images, the pairs of orange dots represent copper ions and also indicate where we believe the active site to be on the protein. The gray clumps in these images also represent where the inhibitor was predicted to bind during the molecular docking process. As you can see in these images, the overall structure of the proteins look similar. However, there is a clear difference between the previous and new docking results. In the previous docking results, the inhibitor was predicted to bind in between two of the active sites, whereas in these new docking results, the inhibitor was predicted to bind closer to a singular active site. This is important because binding closer to the active site would mean a better chance for inhibiting denitrification. On the right side of the slide, you can also see the previous and new molecular docking results for the NOR homology models. In these models, the light blue structures represent heme groups and also indicate where the active site is in this protein. And again, the gray clumps represent where the inhibitor was predicted to bind on these proteins. Unlike in the NER models, the NOR models have large similarity in their overall structure and location of inhibitor binding. On this next slide, the information shown is actually identical to the information shown in the previous slide. However, this information is illustrated in a bar graph. On the left, you can see the bar graphs of the previous and new NOR docking results. These graphs look vastly different, which also mirrors how the NER models look different in the previous slide. On the right side of the slide, you can see the bar graphs for the previous and new NOR docking results. These bar graphs look very similar, which again mirrors the similarity of the NOR homology models that were shown in the previous slide. In order to understand the differences and similarities that were shown in the molecular docking results, we performed alignment of the NER and NOR homology models. As you can see in the NER alignment model on the left side of the slide, the old and new models have an overall similar shape. However, the positioning of the structures is vastly different. This is in contrast to the NOR alignment model on the right side of the slide. Here, the two models have such similar structure and positioning that they overlap perfectly in the image. Not only did we perform molecular docking of fungal NER and NOR proteins, but we also did the same for bacterial NER and NOR proteins. Specifically, we used 65 NER and 7 NOR bacterial models. However, only 3 of each are shown in this presentation. The blue images represent the bacterial NER models, whereas the yellow images represent the bacterial NOR models. When comparing the fungal and bacterial NER models, we saw that the overall structure of both the fungal and bacterial models looked very similar. However, there is a slight difference in inhibitor binding between each individual model. However, when comparing the fungal and bacterial NOR models, we saw that there is a great difference in the structure of these models, and as a result, there was also a great difference in the location of inhibitor binding. Although a lot of information was shared, the results of this research can be summarized in a few key statements. First, in the molecular docking of fungal NER and NOR proteins, we saw that the NER models had similarities in their structure, but differences in their overall inhibitor binding location, whereas in the NOR models, we saw that there was a large similarity in structure and inhibitor binding. These characteristics were understood and verified through the alignment of their homology models. Secondly, in the comparison of fungal and bacterial molecular docking results, we saw that the fungal and bacterial NER models had similarities in structure with slight differences in inhibitor binding location, whereas the fungal and bacterial NOR models had great differences in their structure and inhibitor binding location. Not only is it important to understand the results of this research, but it's also important to interpret these results and understand what they tell us. Therefore, I'd like to go over some of the important results that were produced from this research and explain their significance. First, in the case of the fungal molecular docking results, why is there such a large difference in the NER homology models? One likely reason for this is that the templates that were taken from Swiss model and used as bases for the final homology models in each trial were slightly different. Another important result to interpret is why there was such a large difference in inhibitor binding location in the near docking results. One likely reason for this is that the starting locations for the molecular docking process in the old and new trials were different, which influenced the inhibitor binding location to be different in the old and new trials. Another likely reason is because the molecular docking process is inherently random, which would also influence the results to be different from each other. The final result that should be interpreted is why there is such a large difference in structure of the fungal and bacterial proteins. In the fungal bacterial NOR protein specifically. One likely reason, or the most likely reason for this difference, is that the bacterial NOR proteins have additional functions that are not limited to the reduction of nitric oxide. As a result, the structure of the bacterial NOR proteins will be different than the structure of the fungal NOR proteins. Before I end this presentation, I would like to give thanks to some of the groups and people who made this opportunity a great experience. First, I'd like to thank the University of Georgia's RIU program, who allowed me to have such a great opportunity in the wake of the COVID-19 global pandemic. I'd also like to extend thanks to the other RIU participants who were a source of support and advice in navigating this new experience for myself. I'd also like to thank the Toxicology and Mycotoxin Research Unit, who gave me a lot of valuable information on science and the daily workings of scientists. I would like to thank Dr. Pennerman and Dr. Gold, who were my mentors and helped guide me through this program, and also gave me valuable information on the work of scientists. Finally, I'd like to thank my professors, Dr. Logan and Dr. Olatadoye, who wrote me recommendations and even allowed me to apply and participate in this great program.
Thank you very much, Samari.
Yeah.
And we already have a question for you. And the question was, how do you deliver the inhibitors there's the question, to the fungus in real life? How do you get it into the soil?
Oh, okay. So I believe I should have touched on this, but I may have skipped over it. One of the parts of this research that was important was that we haven't been able to identify the effectiveness of, or how effective each individual inhibitor that was chosen was at binding to the near and NOR proteins. So we need to the only way to really test this is to do it in lab, like in a lab environment. And with the current global stance, we can't really perform those experiments yet. So that's something we have to do going forward. I haven't participated in those or in that discussion for the research, so I'm not very sure. But I do know that that both the enzymes, the near and NOR proteins, and some of the inhibitors are on standby in Dr. Pennerman and Dr. Gold's lab, waiting for that research to start.
Okay, that's neat. And would you just say a word about Fusarium and where do you find it in natural, outside here? What's its? Yeah.
I actually that is not something that I had. That's not information I have readily available to myself.
Okay.
As of this moment.
Okay.
So.
Okay. So thank you so much for the wonderful talk on molecular docking. This was really a fascinating project. And so I will now introduce the next speaker, which is Brittany Howe. She is from a biology major at Clark Atlanta University, and she, during the year, works with Dr. Logan, and her mentor was Dr. Travis Glenn, and they looked at the challenges of looking at microbiome data. And it's my pleasure to turn the floor over to you. Please proceed.
Unmute.
Oh, you need to unmute, Brittany. Yeah. Okay. Just need to, you're fine. Good. You're online.
Alrighty. Thank you. Good morning, everyone. My name is Brittany Howe. My project is on jackpotting. I attend Clark Atlanta University. My mentor for this summer is Dr. Glenn alongside Mandy and Megan. All right. What is jackpotting? We all know what winning the lottery is. We all know what winning the lottery is. It's any game of chance. Jackpotting is the top prize in any game or contest that is typically a large sum of money. And if you guys have your cellular phone or anything, can you guys like pull those out so you could scan a QR code for me really quick? All right. Which color or shape do you think is going to win this jackpot? So if you guys have a phone, just go to camera, scan the QR code, and it will pop up a little survey. So if you guys could do that really quickly for me, I would really appreciate that. If we all are familiar with buying the jackpot, we all know we must buy a spin with the appropriate qualifying bet for a chance at winning. Now I'll give you guys like a couple seconds to go ahead and do that. All right, let's move on. So those spins that you would buy is either a triangle, a moon, or a cloud. What is jackpotting? A jackpot is a large number of amplicons that arose from a single amplicon. Did you buy your favorite shape or color? So this is a visual representation of jackpotting. If you could recall from the previous slide you bought, this is where it comes into play, where you will see the real reason why you bought a spin in the first place. So here you see these small amplicons, which this one is the cloud, the moon, and the triangle. So are you cheering for clouds, moons, or triangle? At this point, we should be focusing on the cloud as it grows rapidly. So from the previous slide, we'll see that, you know, we have less, the same amount of moons in triangle. However, the clouds is growing rapidly. Can you tell who is in the lead? And it's the clouds, as we all can see here. And the winner is clouds. So here, right here, we see the cloud, which is an amplicon from only one specific species. And one cloud, an amplicon is a piece of DNA or RNA that is a source or product of an amplification of events. All of these frequencies, meaning the cloud, the moon, and the triangle, all started in the same community. It's just been diluted as to why we're seeing more clouds. Why is this important, and why are we studying this information? This is a basic problem we have in PCR, where we're taking DNA and using it to make more DNA using a protein acid. PCR is a technique used to amplify a segment of DNA interest or produce lots and lots of copies of DNA. How does the jackpotting applies in the healthcare settings? We use a amplicon sequencing to study variety of built environments in especially healthcare settings. Healthcare settings are important to study as patients within these settings are exposed to variety of microorganisms. This is the healthcare settings and everything right here. This is important because the bacteria in the patient rooms, particularly on the bed rails, consistently resemble the skin microbiota of the patients occupying the room. the bacteria community on the patient's wound surfaces increasingly similar over the course of a patient's stay. And these are all contributing factors of jackpotting. Methods. So to conduct this experiment, we use a mock community, 7 serum dilution, and we use the specific primer V3 and V4 and the PCR machine here, and to create a microbiome analyst using Genius. Results. So this is what we're going to focus on is Pseudomonas dalis. So we're going to focus on that, which is a rod-shaped spiral eubacteria, which is usually motile by polar flagella. All right. So this is dilution 3. Remember, we're focusing right here. And this is 11%. So this, what I'm going to show you in the next few slides is as we go further down in the concentration, we realize that you'll see Pseudomonas dalis just grows rapidly. All right here, this is dilution 4. And Pseudomonas dalis, if you could recall from the previous slide, this is the clouds. This is how the clouds come into play. This is the contaminant. And this right here increased by 26%. Here it's at 57%. It increased by 20% here. And this is in dilution 5. And you're probably wondering why it didn't start at dilution 1, because it's more apparent from dilution 3 to 7. And this is dilution 6. And in this community, they all started at the same point. It's just it's the dilution in the same exact community. And conclusion. Jackpotting is important because we would like to get the accurate representation of microbiomes present, and jackpotting prevents that from happening. It's especially a problem if the masking antibiotic represent microbes that are present. Low concentration of DNA would lead to jackpotting. Jackpotting is important because we could use it to measure the spread of bacteria. And also, for in the future to prevent this from happening, it's an ongoing research. However, the best way we think we could monitor this, the spread of this bacteria, is to create a model where we'll be able to access the environment in the community. And I want to say thank you to everyone. Thank you to Dr. Arnold, Travis Glenn, Amanda and Megan, and Dr. Logan. And thank you to the entire REU team at UGA.
Thank you, Brittany. That was wonderful. So, microbiomes are used very broadly. You know, they're used to examine patients. They're used to examine what's in the soil, what's in seawater. So are there steps that you can take that you avoid this jackpotting problem? Because, yeah.
Okay, so it's still an ongoing research. They haven't reached that far, but the best thing they're thinking that could possibly help, it hasn't been tried out or anything yet, is to create a model where they'll be able to control the environment, and then they could fix it within wherever the jackpotting occurred, they'll be able to correct it.
We had a question from the audience. Were the ones that you were concerned with, pathogenic, bad for us, since you were looking at like Pseudomonas, right? And were those the ones you were focused on in your work?
Correct.
And so you're actually, you think you could be able to distinguish a jackpot of Pseudomonas from the spread in the hospital room? Or
should be able to. With the Pseudomonas dalis, you realize with it on the other the picture of the result, you realize that the Pseudomonas dilis is the only one that's increasing drastically. Like
Okay.
in number. Well, the
Okay.
other.
Okay, and then I have another question for you. What was the reason behind choosing your model organism over others in the microbiome? I guess they're thinking, yeah, that was the question. You focused, or were you focusing just on Pseudomonas?
Yes, I was only focusing on Pseudomonas dilis because it was the only one with the drastic change. That's the only one that jackpotting was occurring in.
Okay. Okay. And that one is antibiotic resistant, I gather, and it's not good for the patient, I presume. No. Okay. Okay. I think I'm just looking for a few more. Do we have any more questions? I don't see any more. Let's see. I guess we're due to have a small break till 11:30. I guess we're a little ahead of schedule. I don't think we'll jump to Kristen quite yet. So if you do have any more questions for Brittany, or please feel free to ask. I've asked my questions. Okay. Okay, so we'll just pause here for a little while and again, if you want to ask questions, all you have to do is type them into the little box below the image, and the speaker will respond. Thank you so much, Brittany. And we're going to come back at 11:30 for Kristen Porter.
You're welcome.
Thank you. Well, welcome to the REU Virtual Symposium. Our next speaker is Kristen Porter. She is a biochemistry major at Mississippi State University, and she has decided to look at a project in which to look at the host side of the COVID-19 problem and look at markers in the human genome that may be related to being asymptomatic versus symptomatic. It's my pleasure to introduce Kristen. Thank you, Kristen.
Hello. Hello, everyone. Like he said, my name is Kristen Porter. So I'm going to share. So my project is about pretty much a genome-wide study on COVID-19. Hold on just a second. I don't want you to see all of my goods just yet. There you go. So, like I said, my project essentially was on doing a genome-wide association study of COVID-19. So, okay. So he already introduced me, but I'll introduce myself again. But hello, my name's Kristen. Here's some background information on me. I was born and raised in Tuscaloosa, Alabama, but currently I've just moved from St. Louis to Chicago, so now I live in Chicago. I'm still a Midwesterner. Like Dr. Arnold said, I'm a rising junior at Mississippi State University. I'm currently majoring in biochemistry, and I'm concentrating that in scientific research while also pursuing a minor in biology. So for this, the entirety of the internship, my mentors and I's main concern was getting familiar with actually being in research and then conducting my studies. So I kind of paced myself. So the main goals within the internship were just for me to properly interpret research papers, learn how to use and get familiar with computational software like Sapelo2, R, Planck, etc., and then to finally conduct the actual genome association study on COVID-19. And then within that GWAS study, I would determine any genetic risk factors or predispositions that make people more or less likely to contract or have different symptoms with COVID-19. And then I was also told to analyze and explain how those specific genes or markers are risk factors. So if I did find anything, how would COVID-19 and that gene interact with each other to create a symptom or a systematic symptom. And then at the end was to learn how to interpret genetic data to reach a conclusion on what I found in the study. So just the two smaller objectives with the research papers, it kind of gave me a written explanation on how to actually conduct specified research, more specifically the GWAS study. It literally pointed out how to do the introduction, how to extract DNA, how to analyze the data. It was a visual representation of how I should conduct my study. It gave me exposure to scientific literature. I didn't read a lot of science papers when I was in school, so it kind of helped me see how other scientists communicate with each other, as well as it kind of showed me within my own research how I can communicate my project to you guys or to other people who aren't necessarily scientific people when I am done with my project, when I was done with my project. As well as by reading all these papers, it kind of made me be more thought-provoking what I was asking or thinking, made me ask questions that I could use to potentially help or influence my own research. And then with the computation software, pretty much the basic goal was to learn how to write code to carry out some tasks. Within that code, I could make and execute extreme mathematical equations, something that might take me 30 minutes to do, a computer can do in 2 seconds. It helped me create plots and charts that I can visually use to present my data, as well as to sort and organize anything that came into my research. Then as well as with computation, I was able to transfer any data among other lab members. That way I could use it for myself in my project or give it to them. But the basis of my internship was my project, which is genome study on COVID-19. But to understand that, I have to understand what is COVID-19. So COVID-19 stems from the virus of severe acute respiratory syndrome, coronavirus 2, or SARS-CoV-2, and that causes the coronavirus 2019 disease. So COVID-19 is believed to come from bats from China. They were believed to be an interspecies virus that transferred from bats and humans, and those bats came from China within their wild animal food market. The first case of COVID-19 was documented in Wuhan, China in December 2019. And with COVID-19, people have a range of different symptoms. It could be something like a regular fever with a runny nose and sniffles and coughing, respiratory failure, which can range from just using oxygen supplementation or actually having a physical mechanical ventilator helping you breathe. Some people have experienced loss of taste and smell. Some have dealt with sensation loss in their hands and their feet, as well as the biggest thing that some people have is no symptoms at all, which is very bad for the rest of the people around, because if you don't have it, we're not going to know that you did not be isolated and be quarantined, and not pass on your COVID-19 diagnosis. So as of yesterday, I checked yesterday, worldwide there are 15.6 million confirmed cases, and of that 15.6, they do have 9.5 million recovered and 632,000 reported deaths as of yesterday for our numbers. So to begin my GWAS study, first I had to figure out who I was going to test and how I was going to test them. So we did 2 different tests. The first test we conducted on patients who were already diagnosed and already found positive for COVID-19, which was around 991 people initially. And we compared them against the total population. Our total population amount we had for this study was around 262,000 people. And then for test 2, we used the same group of patients that were already diagnosed with COVID-19 versus patients who were tested and were seen as negative for COVID-19. So for test one, I think the main thing with test one is that with the total population kind of acted as a control group, you didn't really know if anyone had COVID-19 had been tested for it. And then with test two, my biggest thing that it kind of shows a little bit of a bias. It kind of shows you, like, of those negative people, what kind of advantage they may or may have not had in order to be tested negative or have not be likely to get it, likely to obtain COVID-19. So in order to actually conduct the GWAS study, you have to have DNA in order to look at those genetic markers. So first DNA had to be extracted. Obviously, because of the Rona, we couldn't physically pull blood and pull the DNA out of participants. But what we did use was UK Biobank and their extracted DNA and their DNA in their system to use for our DNA extraction. And so all our DNA extraction came from UK Biobank. The DNA that we extracted came from all 22 pairs of autosomal chromosomes and the sex chromosomes. So we didn't just venture out just for one chromosome, for chromosome 1 or chromosome 2. We made sure we got all the markers on all the chromosomes and see how they play all together. And so after extracting all the DNA, we ended up with 97 million single nucleotide polymorphisms. So essentially, we ended up with 97 million different base pairs for our individuals. But that's a big number. And so if we kept that big number, it would be hard to determine who or what reason why these people would be more or less likely to but have symptoms that go along with COVID-19. So we had to do some kind of quality control. We had to get rid of some of those unnecessary SNPs. So we had to filter them out. Variants like age, sex, genotype array, Townsend index, and other principal components were factored out as the covariants. We didn't have to analyze those genes or gene-specified SNPs for this study. But to go even a step further, SNPs that didn't reach specified scores were also removed. So if the SNPs did not reach the minor allele frequency score of less than 0.1, they were removed. If they didn't reach the Hardy-Weinberg equilibrium law to that's less than 1 times 10 to the negative 6, they were removed. Missingness of SNPs or individuals that didn't reach a score of less than 0.2% or 0.5 were also removed. And then even after all of those, we still continue quality control. So an imputation quality score of less than 0.5. If they had an imputation score of less than 0.5, they were also removed. So doing all that removing was University of Georgia's computation software, which is Sapler 2. Sapler 2 removed all of those SNPs within our study. So 96 million began with increasingly decrease. They continue to decrease as it removed based off of these scores here. And then after we moved the SNPs and we got the SNPs that we deemed as necessary to run the GWAS study, we used Planck and R to create a linear regression as well as find the project itself, the GWAS study itself, threshold for the study. In general, a statistical significance threshold is usually P is less than 1 times 10 to the negative 5, but for our study in particular, our significance threshold is P, which is less than 1 times 10 to the negative 8. And so, which kind of got to our end result. So with removing all of our initial SNPs, we decreased our main patient group, which was the people who were already diagnosed with COVID-19, from 991 to 569 patients in total. Our SNP amount decreased significantly from 97 million to 5.5 million. But with all that being said, we did not necessarily find a significant enough SNP to say, oh, this SNP here is causation, is a marker for genetic disposition to COVID-19. But we did have some that were really close, not a lot, but not really close, so it doesn't really tell us that there's any significant SNPs there for us. But we do have our some visualizations for that. So in this first graph here is the Manhattan plot. And the general threshold, which is that P times 10 to the negative 5 that I mentioned, is right here. And if you look at this line here, you would think that, oh, we have a whole bunch of significant SNPs. But based for our own project in itself, our threshold was this high, and there weren't any SNPs that have actually reached the threshold. Closest one, like I said, was one SNP was 1:6 something, but it wasn't necessarily really close. And then we look at our line of regression. We're doing good until about 6 here, and then things kind of go away. So there isn't really any significance for the first population. And then for the second one, the general threshold once again has some SNPs that are right on the line, but for our GWAS study as a whole, our threshold is a little bit higher, which is literally above the graph in itself, and there aren't any SNPs along. But the closest one that is close to our imaginary threshold line is this RS755 here. And the same thing can be said for our regression line. I'm going to go back a slide. But we started off really well, which just kind of goes in play here. Started off really well up here, and then we kind of just started moving away from the regression line. And the same can be said for the second one here. We started off really well, and then we kind of moved away as we kind of got up the log. So before I do my acknowledgments, one of the biggest things with this is that we didn't find any SNPs to go along with our GWAS study, but we think that we could probably have better results if we did have a bigger population size. Granted, the 266,000 that we did have was decent enough, but in comparison to the millions of people who are confirmed diagnosed with COVID-19, and then compare that to the billions of people on Earth, is a very small population size, and that is not necessarily conducive to the actual population itself. So our research could have been a little bit different if we did have more people to actually test. But that is all for me. I want to thank Dr. Arnold for accepting me into the program. This was my first ever research program ever, so I'm very much new to this. Thank you to NSF for funding this program during the pandemic, because most of the programs said, no, we're not doing this, and they canceled, but y'all came through. And I also want to especially thank my mentor, Dr. Hexon Yee, not Lee, sorry, and his PhD in Yutang Song, because they really helped me, and they were very patient with me, because your girl was struggling. But they were really patient with me and kept me going throughout the project and made sure that I was comfortable in whatever I was doing as I continued on throughout the project. So any questions, comments?
Yes, you have questions. Okay, so where did you get the information on the study participants? In the UK Biobank, that's the first question. The question's probably going to pop up in a second on your screen. There it is. So was it part of the biobank, or was it kept separately? What about their disease status and stuff like that?
I'm sorry. So are you asking me like? I'm confused by your question. I'm sorry.
Oh, okay. Let me try to paraphrase for the person. Did the biobank have both the data about the subject as well as the sequence? So did you go to one place to get both kinds of data?
Yes, I understand now. I understand now what you're saying. Yes, sir. Yes, sir.
Okay. And I would
biobank.
Right. And what I was also curious about, are there other biobanks, from other countries that could also be used, or was this the only one that you had available to you?
This was the only one I had available to me. I'm going to assume there probably is more. But like I said, it's my first time, so I'm thinking my mentor probably didn't want to give me too much to try to handle it one time.
Right.
But that was the only one that I had access to.
Right. And what was the rationale behind setting that very high bar on your markers? It was, it was e to the -8, right? And I mean, because, yeah, what was the rationale there?
Well, I know that I can't say like how it got the number, but I will say when we were starting our study, starting the project initially, one of the biggest issues we ran into reading other papers was that they had the general threshold, and you had so many SNPs that were significant, but they did a poor job of executing or explaining how they were significant. So it's more so like, okay, you have a bunch of these SNPs here, and they all come up as if they have some kind of effect to it. So I'm guessing maybe they made it higher to that way you kind of can get rid of some of those extra,
okay,
I guess, outliers, for lack of better words. But we ran into a bunch of problems with some other papers that we read that were like, how is this significant? And you couldn't find any information as to how even those genes play into your body, let alone how COVID-19 can relate to it. So
I have another question from the audience. There's a recent GWAS in the New England Journal of Medicine. Are you or your mentor planning to try to sort of use that to replicate your findings? So apparently, yeah.
So I'm not sure if they made a recent one here lately, but I know they made one, I think, in the beginning of July, or midway in July. And we did, I did do a presentation with my lab group about it, and they tore that paper up. They were they were very critical of that paper. And one thing about, like I said earlier, about reading these papers is that it makes me think a little bit more. And so when I first read, I thought it was really well, but then hearing other people's thoughts about it was like, oh. But they were very critical of that paper, to say the least. It was funny hearing them talk about it, but they did use that paper as like a guideline, pretty much more so telling me don't do what they did type of thing.
You had another question. Do you know anything about it's going to come up, the function of this SNPs that are the genes that carried the SNPs in your GWAS? Were there any anything associated with O blood type or anything that you could lead you to believe, well, that could be an interesting marker?
For my study specifically, no.
Yes.
The New England Journal one, I did think about that when they brought it up in there, but for mine specifically, no.
Okay.
We didn't have anything significant, so I was like, I don't see the point. I mean, looking for it is not significant enough.
Okay. Okay. So I think we'll stop there and let everybody have lunch. Thank you so much, Kristen. That was a nice counterpoint to some of the other COVID talks, which will focus on the virus. Right? It's nice that we got to look at the other side of the story. So everyone, again, we'll see you back at 1, and Shady Custer will kick us off right after lunch. And thank you for attending, and hopefully you'll see some more really wonderful talks like the ones this morning. Welcome to the REU Virtual Symposium, and it's my pleasure to introduce Shady Custer. She is a biomedical science and major and with a minor in computer science at Tarleton State University. She worked this summer with Dr. Liang Liu, looking at recombination in the beta coronaviruses. And I'm welcome, Shady, and I turn it over to you.
Awesome. Thank you very much, Dr. Arnold. So as Dr. Arnold said, I am going to be presenting my research over the recombination in beta coronaviruses. I have had the wonderful opportunity of working with Dr. Liu on this project. So severe acute respiratory syndrome coronavirus 2, abbreviated to SARS-CoV-2, is a type of coronavirus. Coronaviruses are a family of viruses that can infect a variety of mammals, and they have gotten a lot of attention in the past couple decades because they have been responsible for diseases such as SARS, MERS, the common cold, and the ongoing COVID-19 pandemic. Coronaviruses are further subdivided into alpha, beta, gamma, and delta coronaviruses, and SARS-CoV-2 belongs in the beta coronavirus genus. SARS-CoV-2 is a newly emerged virus, which means that it has low divergence, and a lot of the sequences we currently have are very similar to one another. There are two main ways in which a new viral strain can come about, the first being mutation accumulation, and the other is a process called recombination. And this is where two related viruses will co-infect a cell, and during their DNA replication process, they will interchange segments of their genome in order to create a new viral strain. From a bioinformatics standpoint, recombination can often be confused with mutation accumulation. And the current literature suggests that recombination is rare in SARS-CoV-2, but this is likely not the case, as the sequences are so similar that it's difficult for the methods that we have now to detect it, because when they do recombine, they are exchanging essentially the same nucleotides. So it would just look like mutation. So because they are so similar, my objective was to detect recombination within the beta coronavirus genus instead, since they've had a million years to diverge. And hopefully this will add to the evolutionary history of SARS-CoV-2 and provide information in order to estimate the origins. So luckily, Johnny Chin gave me these gene alignments that I started out with. A whole genome alignment was not feasible at this point in time, so she gave me these gene alignments, and they differ based on the species proportion that makes that up, the number of sequences inside of them, and the lengths. This is fairly difficult to do with beta coronaviruses because they have diverged for so long, so their genome assortment is different. This plot right here is for the S gene specifically, but like I said, they do differ between the different alignments, and the smallest number of sequences that we had was 1,815 in the ORF8 gene, and the largest was 4,101 for the S gene. Since recombination can be confused with other evolutionary processes, we have a multi-step method in order to detect recombination. The first step uses a program called Recombination Detection Program 4, which identifies recombinant sequences, major and minor parents to those sequences, and estimates the breakpoint positions in the alignment. From that information, we wrote an R script to break the alignments at the breakpoint and create phylogenetic trees for either side of that breakpoint. And so in the phylogenetic incongruence test, this was done through RAxML using the PH85 test to see if there was a significant difference in the topology. If there was, that indicated that there was recombination, and that event would move on to the third step, pairwise identity. And this is just a plot that shows the identity between the major parent sequence, the minor parent sequence, and the recombinant sequence. And you can compare them throughout the entire sequence. So RDP4 initially detected 824 sequences that showed evidence for recombination. Of those 824 sequences, there were 103 unique recombination events. And this is kind of just a picture of what that looks like. Of those 824 sequences, the phylogenetic incongruence step decreased that data set by 81.8%. And so this is an example of an event that would have passed this test. And this is because for the major sequence of that recombinant sequence, the recombinant is most closely related to the major parent sequence. And then for the minor sequence of that entire recombinant sequence, it's most closely related to the minor parent. And so this just shows the difference between on either side of that breakpoint. And then for those that moved on to the pairwise identity plot, we had a further decrease of 86.67% of our data set. And this is just a good example of what recombination looks like on this plot. This pink area right here is the recombinant region within the recombinant sequence. This yellow line that you can barely see down here at the bottom is the identity between the major and minor parents. This blue line is the major parent versus the recombinant. And so you can see outside of the recombinant area, it's very high identity, but as soon as you go into that recombinant region, it drops down to 0. And the opposite is true for the minor parent compared to the recombinant. And that's exactly what we're looking for with this recombination. And so overall, from step 1 to step 3, we had a 97.57% reduction in our original data. And so you can see in this plot right here just how dramatic that reduction was. And this just speaks to the importance of having this multi-step method, because RDP4 just grossly overestimated the amount of recombinants within our data set. So we ended up with 20 sequences following the 3 steps. Of those 20 sequences, there were 5 unique events across 3 different genes. And this table right here lays out the 5 unique events that we have. So they were between the M gene, the N gene, and the S gene. And some important recombination events that I wanted to point out were right here in the N gene. There was a SARS-related virus that recombined with a SARS-related virus to produce a SARS-CoV-2 virus. And then in the S gene, we had a Rhinophis bat beta coronavirus recombined with a SARS-related virus in order to produce a SARS-related virus. In my abstract for this presentation, I did state that I had a SARS-CoV-2 recombination and a SARS-CoV-2 recombinant from the bat coronavirus. Unfortunately, those events did not pass all 3 steps, and so I felt the need to say something about it here. I don't want to mislead people, and so I just wanted to address that. So just because we didn't find direct evidence of recombination occurring in SARS-CoV-2, I just wanted to remind everyone that these sequences are still very similar. And so likely the methods just couldn't detect recombination because the identity is just too high. So recombination has occurred in beta coronaviruses. The events that I have talked about within those genes, there have been other literature that has shown recombination within those genes as well. And so it has occurred in beta coronaviruses. And so as far as I know, there's no reason why it should not also occur in SARS-CoV-2. And this is important because the pandemic is ongoing. And so if we can get information about recombination in the past of SARS-CoV-2, then we can apply that potentially to predicting changes in SARS-CoV-2 in the future and ultimately in COVID-19. And then researchers who are using RDP4 should be aware of the overestimation of recombination by RDP4. And in the future, we do plan to investigate the between gene recombination instead of the within gene recombination, which this project mainly focused on. I would like to acknowledge Dr. Jonathan Arnold, who is the program director for this REU and who ran this program and continued it in a way that was very helpful to us students despite the circumstances that we were in. I'd like to thank Johnny Chen for providing the gene alignments that I used and for providing that plot that I spoke about earlier. The National Science Foundation for providing funding for this project and this program. And then my COVID-19 research group that I met with weekly. I learned a lot from you guys, so thank you. And then just for all the friends and family who have supported me along the way. Are there any questions?
Thank you very much. You have a question now from the audience. Do you think recombination could, and the question's going to appear, do you think that recombination will make COVID-19 a worse virus potentially, or said another way, could COVID-19 recombine? Well, a follow-up question is, could COVID-19 recombine with viruses that are not closely related to it?
So recombination, in order for, because it is a type of mutation, and so in order for that virus to survive, when it recombines, whatever it gets versus what it gave, that needs to be functional. And so really that would just depend on what it gave and what it received. As far as SARS-CoV-2 goes, you asked about whether I thought it might become more virulent or more dangerous. I believe that this is more relied on, or it depends more on social distancing procedures. So there's this trade-off in viruses between rate of transmission and rate of fatality, like how dangerous the virus is allowed to become. And so if it's a very fatal virus, then it will kill its host before it can be passed on. And that causes, so it can't be transmitted as quickly. But if it's not as fatal, then it'll be able to be transmitted a lot further. And so social distancing kind of helps to prevent those more virulent strains from becoming more widespread. I mean, it's possible for that to occur, but if these measures are kept, then they shouldn't become the main strain.
Okay. And what are some of the effects of recombination on the gene trees that the group's working on within say, SARS-CoV-2?
Within SARS-CoV-2?
Yeah, as an example.
It's difficult to say because those sequences are still very similar, but I'm not quite sure what happened with the Zoom. Can y'all still see my presentation?
Yeah.
Okay. Oh, there we go.
Let's share it again. Yeah. It should be coming up. There it goes. Yeah.
So, of course it's not letting me. Okay. So this is like an example of how it affects the phylogenetics of the sequences. And of course, this is broken at the breakpoint. So if you're looking across gene trees, then you would see maybe in one gene, this specific sequence falls out with these groups of sequences, like group A, for an example. But then in maybe one or two genes over, it's going to fall out with group D. And so that can mean a few things, but one of the conclusions to that could be recombination.
Right. Okay. So there's one other question that person in the audience had is, how distantly related can the beta coronaviruses be and still recombine?
That is a very good question. Unfortunately, I don't have the answer for that. Okay. I'm sorry.
Yeah. Okay. Well, thank you very much, Shady. And we're now going to continue this story with Grant Wong. He is a biochemistry major, and his work has focused on the spike protein in I believe the beta coronaviruses. And I'll turn it over, and he's going to be looking at this spike protein, which causes virulence in, for example, SARS-CoV-2. Go ahead, Grant.
All right. So, can you all see my screen?
Yes.
All right. So, I performed this summer a phylogenetic analysis of the OC43 human beta coronavirus. So now coronaviruses are a real diverse group of viruses that infect all sorts of animals, humans, bats, rats, and a lot of different types of mammals. And so they're split into four main groups: alpha, beta, gamma, and delta. So out of the beta coronaviruses, there are 5 main ones that infect humans. In lineage A, we have the OC43 and human coronavirus 1. In lineage B, we have SARS-CoV-1, which was the original SARS, and then SARS-CoV-2, which we're all so worried about right now. And in lineage C, we have MERS coronavirus, which causes Middle Eastern respiratory syndrome. Now, the OC43 that I'm going to be looking at is one of the viruses that is a cause of the common cold in human beings. And it has a lot of similarities to other types of beta coronaviruses and how it's spread. It's a cough or through touch transmission. And if you look at the diagram right here on the right, this is just a generic coronavirus. And these little pink protrusions are called spike glycoproteins, and they're the source of the coronavirus's name. And these spike glycoproteins are real important for the virus because they're what attach to a cell's receptors and allow the virus to gain entry. They're also the main target of antibodies. So the shape of these spike proteins determines how well the virus is going to be able to enter a cell and also how well it's going to be able to evade the immune system. So that's why we're specifically looked at the S gene of the OC43 virus. And for phylogenetic analysis, one of the major parts is creating a phylogenetic tree. And a phylogenetic tree just shows the evolutionary relationships between different organisms, maybe they're species or maybe they're different strains of a single virus. So at the very beginning of the tree, the oldest part, we have the most recent common ancestor of all the different taxa of the tree, branches right here. And whenever a branch splits apart, it's called the node. And all at this end of the tree is the closest to the present. So a tree doesn't actually definitely tell you, hey, 2 organisms both evolved, from this ancestor. It just says if 2 different organisms are grouped closer together on the tree, that means that they're more closely related. This used to be through things like, oh, these 2 animals both have red fur, so they're going to be grouped closer together. But nowadays, it's based on genetic similarity. And this just helps us show how different strains of virus across the world are related to each other and predictions that we can make of these viruses as they continue to evolve in the future. So this is a linear gene map that I made, and it's just to help illustrate how these viruses change over time. And this is an illustration of the entire genome of a few different beta coronaviruses that I mentioned earlier. So the first one is the one that I looked at, which is the OC43. And you can see the S gene right here in red is closest in size and location to, I suppose, another coronavirus, another beta coronavirus that's in the same subgroup, which is HKU1. These 2 are the most closely related. As you can see, the S genes are the closest, and it continues to change. So as these things evolve, their genomes change, the locations of the genes change, and all that. So it's important to look at it from a phylogenetic standpoint. So the data that I used was downloaded from NCBI, the virus database. I searched for OC43 whole genome sequences. So I searched for sequences that were over 34,000 base pairs in length. And I got 178 whole genomes that were sampled from across the world, and they were collected from 1983 to 2018. And so first off, with the raw whole genomes, I had to align them first so that they could be compared. And that alignment was done using a program called MAFT. And then a software called AliView was used to visualize the alignment and to process it afterwards. So here's a screenshot of what the alignment looks like. You can see all the different nucleotides are lined up to the best of the software's ability, and any little differences, these little differences aren't going to mess up the alignment, but a large difference between a single genome and the other genomes can really mess up the alignment. So any sequence that had that was causing a large gap in the genome, that was removed. And in the end, out of the 178, about 47 sequences which were removed, which sounds like a lot, but I'll be going over that later. So then after the alignment was completed, the S gene was located on the genome and then extracted. And then a maximum likelihood tree was built from the S gene data. And maximum likelihood tree was produced so that we could get an initial look of what a phylogenetic tree looked like and also remove temporal outliers. So a maximum likelihood tree, I think as Vibot said earlier, a maximum likelihood tree is a tree built, its likelihood is assessed on how similar is it to a model, how well does it conform to a previously established model. So once that tree was built, it was analyzed using a program called Tempest, and any outliers were removed. But here that wasn't necessary. There were no significant outliers, so none were removed. And then once we've got our trimmed down S-gene sequences, they were put into a program called BEAST. BEAST is a Bayesian phylogenetic analysis software that assigns posterior probabilities using Bayes' theorem to all the different trees and uses a Markov chain Monte Carlo MCMC algorithm to sample the different trees. So then we looked at the MCMC trace using a program called Tracer, and I've got it right here. And basically what this says is that we ran our program for enough iterations that it worked basically. This is showing that the MCMC chain has converged. This is the trace of the MCMC chain. And so then, all right, I think I skipped a slide there. Then after we've made sure that our data is all good, then we built an MCC tree from the trees that were sampled with BEAST. So unlike a maximum likelihood tree, an MCC tree looks at all the different clades within a tree and assesses which tree has the best clades, the most frequently appearing ones. And this is the final tree. So I've got different samples from different nations. I've assigned different colors to them. So US is going to be in the blue here. China is going to be in the green, Malaysia in the red, and a few other countries. And what this tree really is able to show us is that, I'll go back to the tree, the nations are typically grouped together with some exceptions, but the nations are typically grouped together, which means that the virus tends to evolve and diverge within nations before it goes on to spread to a different nation and then on to a different nation. And another conclusion that we found was that if you look at this clade right here and this clade right here, even though all the samples were acquired relatively recently, around 2016 for these, they diverged. The time that they diverged is all the way back in 1985. So that's a difference of about 30 years. And it doesn't mean that these viruses have just been hanging out for 30 years, not changing whatsoever. It just shows that even though these viruses are relatively new for these two clades, they're genetically most similar to these older samples. And so in the future, the same methods that I used to look at the S gene of the OC43 virus can be applied to different genes and different viruses. And also conclusions that I got from the OC43, it doesn't just have to be used for the OC43, it can be used for other beta coronaviruses like the SARS-CoV-2 that's going around right now. And another thing that I wanted to add that I mentioned earlier is that during the alignment step, when I was looking over the alignment, I mentioned I removed about 47 sequences. And the reason that they were all removed is that they just didn't align very well. They prevented a proper alignment. And so one of the big reasons I think why is that it just had to do with the data itself. All those sequences came from the same lab at around the same time. It just might have been the way that they sequenced the genome that this is why that happened. All of them had to be removed. But regardless, the same methods used for other viruses, other genes, and by looking at one virus, you can look at similar viruses. So finally, I'd like to thank my mentor, Justin Ball, for really guiding me through this process. The graduate students, Johnny Chen and Lambodar Damodaran. You know, when I was starting this program, I knew virtually nothing about anything that was going on. You know, step by step, every week, they just helped me move along, move through this. It was really great. And then Dr. Jonathan Arnold, of course, you know, putting together this program, even in the face of all this adversity, bringing us close together. So thank you very much. Oh yeah, and of course, generous support from the NSF. All right.
Thank you, Grant. We have a couple of questions from the audience. Could you elaborate on some of the exceptions to nations grouping together? And how does knowing about the evolution of the virus help us to fight the disease? So you got one very specific question and one broader question.
All right. All right. Well, I'm going to share my screen one more time. And
okay.
So I think what, with exception, is that if we look at these two groups of different United States samples, both in the blue United States, it shows that, you know, they're kind of separate, which means that whatever traits were present in the S gene for these viruses is carried on, you know, in these samples up until modern day. Whereas these samples over here, which are both from, this is all from Tennessee, this is all from the United States, all from about the same time period, same labs, these are in a completely different part of the tree, which shows that even though these two groups are from the same area, same region, same virus, they're way less similar to each other than they are to other viruses. And would you mind reminding me of the other question one more time?
Okay. Well, actually, I've got there are three. Oh, here it comes. How does knowing about the evolution of the virus help us to fight the disease?
Well, I think I might have glossed over this kind of in the beginning, but as we look at the virus as it changes from region to region, it's really useful for helping us to predict where it's going to go next potentially. And also, you know, because if we look back at the tree, we can, if there was a pattern such as it oftentimes went from the US to South America to US back and forth, then a phylogenetic analysis can help us to predict, hey, if it's in the US right now, it might go to South America next. So it's valuable for predicting trends in the future. And along with the data of the genes, I think I showed the gene map in the beginning, which shows how the genes rearrange themselves as the virus evolves. You know, looking at the tree itself, looking at the phylogenetic analysis, it's a big part to be able to predict how the genome changes.
And I think there was one more question after that. Let's see. Here it comes. How does knowing about the evolution, well, let's see. No, it's the next one. Do you think your result, oops, do you, here it goes. Do you think your results suggest that the S gene protein represents a relatively stable target for vaccine development, or it's not a good target? Or wait, yeah, how stable is it as far as coronavirus, beta coronaviruses?
Well, within the OC43 virus, which is what I looked at, based on what I've seen over the course of the last 40 years, which is what the data is from, over the last 40 years, you know, the S gene definitely changed, but relative to some other viruses, it's relatively stable. Of course, compared to SARS-CoV-2, it evolves faster, but the S gene is relatively stable. But you know, between species, you know, it's real different.
Okay. And then another question from the audience was, what does MCMC stand for? So.
Oh yeah, that stands for Markov Chain Monte Carlo, which I'm sure Dr. Arnold can go into this way better than I can, but it was basically the way that the software, that the program Beast was able to sample the best trees from all the trees that it generated.
That's a pretty good description. Okay. So thank you, Grant. That's wonderful, wonderful talk. And it's now my pleasure to introduce Lily Stratford Adams. She's a biology major at Doane University. And she's looking at networks too. It's just root networks. And she's going to look at the problem of trying to get at the genetic basis of root length and diameter in real roots. Okay. Thank you, Lily. Oh, and Alex Bush is her advisor.
Hi, can you hear me?
Yes, we can hear you.
Okay. Hi, I'm Lily Stratford Adams. I'm a rising junior at Doane, and my summer RAU project was image processing with root architecture. And I worked with my mentors, Momo Shi and Dr. Alexander Bush, and corresponding mentor, Dr. Tessa Durham Brooks. Oh, sorry. How do I do this? Okay. So, sorry. So genotype refers to the hereditary genetic information that an organism expresses, also known as our traits. Oh, can you see this? Not every genotype is physically expressed, but there are those that are, and they're called phenotypes. And different phenotypes make it possible for us to visually see genetic variants. So phenotyping is the process of predicting an organism's phenotype based off of only genetic information. And while there have been vast advancements to improve efficiency, cost, and ability to conduct genotyping on a large scale, phenotyping has not received the same treatment. And large-scale phenotyping on an accurate level is limited and often requires a lot of human input. And if we were able to do this automatically, we could analyze the phenotypic consequences of different genotypes much easier. So my research pertained to finding associations between phenotype and genotype, looking at images of roots to extract the traits of root length and diameter using image processing techniques. I analyzed 1,440 images of plates, and this included 6 sets of 48 plates over 5 days, and altogether it covered 252 ascensions of Arabidopsis thaliana. And Arabidopsis is a plant well known for its well-annotated genome and large natural variation. It is a model organism used to study basic biology. This study is actually a revival of a previous project by Dr. Radka Slovak, who published a paper in 2014 where she and her lab developed an image processing pipeline to gather and analyze those 1,440 images of root plates to find associated genes for different phenotypic traits, including length. However, no associated genes were found. My mentors and I believe that the lab did not find any genes because the image processing pipeline, which used a machine learning algorithm to detect the root, was flawed. And here you can see that the algorithm called root length starting at these points, but in reality, it really should have been called at these points. So there were also some other issues, including the script having a 21% false positive rate without human checking. And so the logical thing to do was to develop our own image processing script, and we did this in Python using the NumPy and OpenCV libraries. So image processing is a subset of computer vision where images can be computationally analyzed using algorithms to help detect and measure areas of interest. And we wrote scripts using different approaches than previously used, using general processing techniques to extract the medial axis of each image in order to get the trait values. So the medial axis is defined as the collection of points within a polygon that are closest to more than one of the edges. And so in this picture, we can imagine that the blue polygon shown is our image, and the vertical red lines that are going downwards are our medial axis. And in my script, we took the image, we grayscaled it, and then we pulled out the individual roots using connected components, and then extracted the medial axis. All of these processes were made possible by the functions used within the OpenCV library. We also addressed the issue of underestimating the root length by only deleting the leaves, which you can see here. So the script output a lot of data, including the values of length and diameter for every single root. I then wrote a Python script to assign the root a gene using the accession index. And I extracted the important parts of the columns and associated them with each other. I then collected the mean diameter and length for every day and that we would use to run the genome-wide association study. And the script utilized pandas and CSV libraries to help reorganize the data frames, automating something that would have been very tedious to do manually. So a genome-wide association study, or GWAS, was conducted on our trait values. And a GWAS is a statistical method for correlating single nucleotide polymorphisms, or SNPs, with one or more phenotypes of interest. And SNPs are single base pair changes in the DNA sequence and are used as markers for genetic regions. And the way that this works is that it looks for associated genetic variations and then runs an odds ratio based off of those with the genetic variation and those that do not. And it will then pinpoint the chromosome where the variation is considered significant, which is denoted by the SNP passing a certain threshold line, which is where my arrow is pointing to. And so the big part now is to see if my scripts actually did work. And we fed our extracted length and diameter phenotype traits into EasyGWAS, which is an online platform used to conduct genome-wide association studies. And the great thing was that we found 2 matches for 2 genes. And the first one was one for diameter, which was a hexokinase that phosphorylates glucose, which was found on day 2 of the images. And then for length, we found WSD1, which is a wax ester synthase. And those are some Manhattan plots, and you can see with the green line or with these blown up images, the red dotted line, those are our thresholds. And the areas of the SNPs that we found interested that correlated with the gene are the pink dots. So I guess some takeaways from this whole experience was that I improved my problem-solving skills. I got to experience the researcher lifestyle, and I really enjoyed that. Future work that I could do was probably to improve my scripts and the pipeline process itself. The images that are shown here are some of the, not errors that I found, but some of the parts of my script that I had to troubleshoot, and I just thought that they were funny. And something really exciting is because we had the 2 genes that were found associated with the traits diameter and length, we can now do in lab experiments with them. So yeah. So thank you very much for listening to my presentation. I would like to acknowledge my mentors, Momo Shi and Dr. Alexandra Bush and Dr. Tessa Durham-Brooks, also Dr. Arnold for coordinating the program. I would also like to acknowledge Dr. Bush's Computational Plant Science Lab because all of them were very supportive of me, and the NSF for accepting me and allowing me this opportunity, and also my parents because they've been very supportive of me this whole time too. So thank you.
Great, wonderful. You have quite a few questions. I'll just start with the top. Did you actually ground truth your measurements that you derived from your pipeline, and how did they compare to that previous work which didn't find anything? Were the measurements very different? I think you indicated they were too short or something.
Some of them were too short, and there was an underestimation of the root length. And so we looked at 252 ascensions, which was, they looked at 178 because theirs didn't work that way. With the ground truth thing, I think there could be further research in it, but I think it works.
Okay. Let's see. So that if we could just go to the next question, which is, did you have or collect? Oh no, that's the one we just did. So there should be the next question. Here it comes. How do you think, how much accuracy was your, what was the accuracy of your pipeline compared with the original published pipeline? Were they different? You've indicated there's a bias in the length. What was the accuracy like?
So I did not calculate the accuracy of my method. However, the genes that we did find, actually, there's potential to make sense of why they would associate with the diameter and the length traits. But I do think that when I said in my future work that I should reevaluate my script so that it could have a higher accuracy.
Okay. And we've got some more questions. The next one will show up in it. Okay. Does the image processing allow you to tell what genes are being expressed?
So what we did was we kind of did a work backwards method where we took so there was all this data that was provided, except for it was almost a double checking kind of method. Yeah.
Okay. So, but there was expression data on this data set. Okay. So that you could look at that. And then I think there's at least one more question. This question is, were the identified SNPs that were significant located within the gene coding sequences, or were they in regulatory regions?
So I will be honest, when we did the GWAS, I got the results from that with about 3 or 4 days ago. So I think that I would need to analyze that a little bit more.
Okay. Okay. I'll ask a last question. Are you planning to write up your software and put a paper together on it?
I would really like that, but I think that I would probably have to revise the code a lot.
Okay.
And
okay.
But I think that there is potential for that.
Yeah, good. Thank you very much, Lily. That's great. So our last speaker is Isaac Reinhard. He's chemistry with a minor in CS. Or did I get it backwards? I think that's what I got written down here. So he goes to I think, let's see, the University of Portland. And what he is going to talk about is visualizing gene trees of SARS-CoV-2. And thank you. Please go ahead, Isaac.
I'm going to share my screen.
Okay.
Oops.
Yeah. There you go. Just go full screen there.
All right.
Great.
Hello. As Dr. Arnold said, my name is Isaac Reinhard. I work with my mentor, Michael Scaro, who is a PhD student, and also a Dr. Arnold as well. And my project was on visualizing gene trees of SARS-CoV-2 geographically. So just a quick overview on SARS-CoV-2. So it is the virus that causes the disease that we are all accustomed to by now, known as COVID-19, which to date has infected or has spread to over 206 countries, infected more than 16 million people, and caused over 600 deaths. So now the background on more genetic background. So it is a ssRNA virus. The genome is roughly 29.2 kilobases. It contains 4 genes that encode for structural proteins, the S, M, N, and E genes, while the remaining genes encode for accessory proteins. Further, interesting to note, the N protein is the one that holds the RNA genome, while the S, E, and M together create the viral envelope. And then the S protein, which is the little red spikes on the virus, is what allows the virus to attach to the host cells and infect them. So in this research that we conducted over the summer, we used a phylogenetic approach to study the evolution of the SARS-CoV-2 sequences. So phylogenetics allows us to study the evolutionary relationships, in this case between genes of interest. We can construct these trees based off of the genetic sequences, which allows us to infer evolutionary relationships and how the sequences of each gene has changed over time. So the components of a phylogeny really quickly are its topology, which is its overall structure, the branches, which indicate the amount of genetic change from a branch point. Nodes are, of course, at the ends of the branches, and the root is the origin of the tree. So there are different ways to root a tree, and a common approach is the outgroup method, where you include one or more sequences that are distantly but also closely related to your sequences of interest. And that's how you are able to root the tree. And then lastly, the confidence or the support values for each branch, and how confident we are that they're actually showing meaningful change. So then moving on to kind of the pipeline that we used. So we started with our question, which was, are there mutation rates different in the genes within the SARS-CoV-2 genome? We believe there are, and that this can help in constructing more accurate groupings of virus strains than what has previously already been done. So with our question, we then had to collect our sequences, align these sequences, and then estimate trees through a number of different programs, and then estimate the error from these models. So moving on to our sequence alignment processing. So we use the database known as GIS-Aid, which is the global initiative for sharing all influenza data. We downloaded around 8,000 sequences from this database and aligned them using MAFT, with the shortest sequence length being around 300 bases and the longest sequence length of a gene being 22,000 bases. However, even though there were around 8,000 sequences, you can see in the table next to the workflow for each gene, there was only a certain amount of unique sequences, which Shady kind of alluded to, showing that a lot of the sequences are identical to each other, which physically makes sense just because of the small time course this virus has been able to mutate. If you'll notice, the genes where there's a smaller number of unique sequences are the smaller genes, whereas like ORF1A is the biggest gene and therefore also has the most unique sequences. So from these aligned FASTA files that we had, we then had to screen them to contain only unique sequences, because many programs don't accept duplicate sequences. So for sequences that had many different strains showing that same sequence, we constructed a group based off of that, where we just identified it with a unique number and the composition or how many strains shared that sequence. I will note, which I'll kind of allude to at the end of my presentation, in these groups differed across each gene, meaning that certain strains could share sequences with a different set of strains in each gene, which was kind of unique to see in our analysis. So now that we had our alignment files, we then decided to use RAXML, which stands for Randomized Accelerated Maximum Likelihood, as a program to construct our phylogenetic trees. It is used for phylogenetic analysis of large data sets, using a maximum likelihood analysis. It also calculates bootstrap values for these branches, and allows for you to use different substitution models. So for our parameters, we use GTR gamma substitution model, 100 bootstrap replicates. We used a multi-threaded version that ran on the supercomputer at Georgia, and we rooted the trees with bat genome sequences. So as I kind of was discussing, so for these groups that contained many different strains sharing the same sequence, we wanted to have a representation of the geographic regions represented by these groups to see their composition. So we just focused on kind of a continent-based system where you can see different colors corresponding to different continents. In the future, it could definitely be refined to have more specific areas, but those were the ones that we decided to choose. So these would be linked to the specific group nodes. So now that we had our trees constructed using Raxamol and these ribbons, we were able to put it all together. So we decided to use a toolkit in Python called ETE that helps visualize trees. So if you'll see, these are circular trees rooted in the middle. For the nodes, they're either a sequence if it didn't share its sequence with a different strain, or the groups corresponding with their geographic ribbon. There's also displaying the branch distance and their support values. So these are two genes that had kind of an interesting composition going on. So in this ORF8, we were able to identify two distinct clades. So the green clade contained all the sequences that actually came from Australia, whereas the blue clade was primarily North America and Europe sequences. You also notice that sequences from Asia are distributed throughout the entire tree, which makes sense because this is the geographic origin of the sequence. However, as I alluded to before, you'll notice that there were some North America sequences that were in both clades. So one thing that we could do in the future is to show which of these were from the West Coast versus the East Coast. Would be something interesting to look at, because it has been proposed that the viruses from the coasts come from different areas, with the West Coast primarily coming from Asia versus the East Coast coming from Europe. So that would be one thing to look into the future. And then in the gene N, we also found two similar clades, with the orange being similar to the green clade and the yellow being similar to the blue clade. So these were two genes with that kind of shared a similarity. One thing to note is, as I was saying, that we rooted these trees with the BAT gene respectively, and in the ORF8, so that the BAT is the dark pink. It's kind of a little hard to see. But you'll also notice above it is a light pink, which corresponds to the pangolin sequence that was included in these alignments. So you can see that these were both found to be genetically distinct from the other SARS-CoV-2 sequences, whereas in the gene N, while we found the BAT to be genetically distinct, the pangolin was actually grouped with the SARS-CoV-2 strains. So that was one interesting difference that we found. Here's another tree that we had, but this one actually it only showed a genetic divergence between the SARS-CoV sequences and the BAT and pangolin. So there wasn't really a genetic distinction between the SARS-CoV-2 sequences to contrast the previous findings of other genes. And one gene that unfortunately we weren't able to get through was the S protein, which would definitely be one we want to look at because it's responsible for infecting host cells. So that would definitely be an interesting tree to look at in the future. So hopefully that will come back so we can look at that. So overall, our preliminary results indicate low mutation rates across these genes, but there were differences present between genes. To validate these results, we would need to run more bootstraps. One thing is we set the bootstraps that we ran, but there's also a method I recently found that you can have Raxamol estimate the number of replicates it needs to do to have a valid results and accurate results, and also solidify the workflow for these alignment files. So as we kind of found, we had a few problems with our alignment files in the beginning of the data processing, and ultimately we just wanted to make sure that our alignment files are very accurate because they are the basis of all the trees that we construct. So in the future work, these gene trees will be used to annotate non-synonymous versus synonymous mutations in the SARS-CoV-2 genome. These will also be used to help construct a gene network, which we can then make viral groupings based off of this network to have a more informed awareness of how these strains have evolved. And then, as I alluded to with the groupings, we noticed that different strains grouped differently within each gene. So that could be a seed to investigate potential recombination between genes in the SARS-CoV-2 genome. But again, there was no analysis done on that, but just the initial groupings suggested that could be something we would want to look into. So overall, thank you to my mentor, Michael Scaro, Dr. Arnold, Dr. Liu, and many others in the COVID-19 group at UGA. They made this experience very helpful, and I learned a lot, and very thankful that it was allowed to continue. So with that is the end of my presentation, and thank you for listening.
Thank you very much. So which gene was it that had you had three groups in it, right? Was it the N? No, yeah, one back. I guess it looks like, oh, my eyes aren't good enough.
Yeah. In PDF version, you can kind of zoom out.
But there are three groups, and the one that I saw, that's much better. So it also looks like that there's a spatial coherence in the group, in the tree itself, right? Because the blue is those are the U.S. populations. Am I?
Yeah, North America.
Yeah, that's North America. So that'll be, and then the yellow is Asian throughout, so that's spread out. And what are the red ones there?
Red is Asia. The yellow is Europe.
Oh, yellow is Europe. Okay. So it'll be very interesting. You are getting differences between the genes, which is a good argument for an integration tool like the one that Vaibhav talked about, that this will be the basis for creating a species tree for SARS-CoV-2. Yeah, so that's great. And the Pythons, you had a Python library that you used to do the strips in this diagram. Does it give you other ways of organizing the spatial data as well?
Are you talking about how to organize the tree itself?
Well, for example, can you? I'm wondering if it gives you other visualization.
I decided the circular because it could fit on one screen.
Right.
I had also a rectangular mode, except they're kind of big to show on a screen.
Yeah. I like the circular one. I think that's much better. And I'll finish with one more question. Do you think each of these sequences have a time of collection, right?
Mm-hmm.
And I'm just wondering, on Grant's trees, we had a time scale. So I'm curious if we can put a time scale on these trees as well, using that time of collection, maybe.
Yeah, they were talking about that. We could put these in the Bayesian analysis to get a date. Of course, it'd be over a much shorter time.
Right. Okay. Well, thank you very much. And this, I think we're ending right on time. And we thank the audience for attending, and I hope the all the speakers will just stay for a moment. We're going to follow up on Lily's suggestion, just to have a huddle about what we can do next year a little bit better. So thank you all for coming. Thank you for the family support. Thank you for all the mentors. Thank you to Sally for paying for everything. And thank you for Dean Dorsey for providing support. And thank you for Clark Atlanta providing the support over the many years for this program. And all the contributors. There are a lot of people that went in to make this program successful. And I'm grateful for their support, including our partnering other REU sites. Thank you.